Good morning everyone on shore, or whatever time of the day or night it is in your part of the world. This is Schmidt Ocean Institute research vessel Falkor and ROV SuBastian, and we are live from the Central Great Barrier Reef off the coast of Queensland, Australia. This is the second dive in our current northern depths of the Great Barrier Reef expedition. My name is Marty McNeil and I'm a marine geoscientist from the Queensland University of Technology in Brisbane, Australia. We currently have 3 scientists on board the vessel at the moment. I'm joined by Dr. Luke Notiff, also from QUT in Brisbane, and Vicky Lowe from the University of Queensland. We also have a group of collaborators and scientists that are joining us remotely onshore. And our dive today is at a location called Bowls Slide, and we're situated approximately 68 nautical miles just northeast of Townsville. We are on the outside edge of the Great Barrier Reef, and at the moment we're in about 230 metres of water, and we're sitting at the bottom of a steep slope which is a scarp and it resulted from an undersea landslide that happened here, we don't know exactly when, sometime in the geological past, and we're going to make our way from the bottom slowly up the slope face and through the mesophotic zone, which is the twilight zone of the coral reef, and we'll be ending the dive. Hopefully we'll get quite shallow today, shallow for ROV SuBastian. So what we're looking at the moment is we're on the seafloor at the bottom of the slope, and we're looking at some reef blocks, which are large boulders that have come down as a result of that undersea landslide.
That's nice.
And that is very nice. So these reef blocks give us a hard substrate, a surface for other organisms to colonise, so they become habitat. And so we're interested in all sorts of things because we've got quite a broad range of scientists that are involved in this expedition. We've got people that are interested in the geology, the rocks and the sediments, and then of course the biology. So these hard substrates form habitat, and we have coral biologists, ecologists and taxonomists involved in our team. And just before we went live, unfortunately you guys missed it, but we had a couple of sharks hanging around, but they seem to have left us now. So we're just going to have a little look around here for a little bit and see what we see. And then we'll start heading along our transect. There's like a couple of fish in the background there. We might slow down around this boulder here and take a look at these corals that are on this rock here. I've got our colleague Jeremy Horowitz sitting at home onshore in Townsville, and he's messaging us at the moment with some interesting things that he'd like us to zoom in and take a look at, and that would be the corals that are on this rock right here. Maybe we have some fish people joining us on the YouTube chat as well. Looks like we've got a little mermaid's purse on that attached to that coral there.
Shark.
Yeah. It's a shark egg sac. We call it a mermaid's purse. Is that what you guys call it?
Vertical.
Yeah.
Shark egg. Yeah. I like mermaid's purse. Shark Mama giving shark tourists.
Looks like we've got Jeremy joining us shortly.
Yeah. Can we verify that it's being broadcast on YouTube?
Jeremy sailed with us on the previous expedition to the Queensland Plateau coral reefs. Unfortunately, Jeremy wasn't able to join us on the current expedition, but he's certainly here with us in spirit, and I know that he'll be tuning in and watching every ROV dive. Hey Jeremy, I can hear you. I do.
Let me focus on one of them and try to zoom in.
Yep, we can do that.
So on the last expedition, we collected obviously a lot of black corals, and some of them turned out to be a species that hosts a worm that modifies the skeleton, and now we're interested in sequencing the entire genome of not only the coral host but the worm species that live inside of it to understand how they have co-evolved. Where we don't yet have the people on board to snap freeze the tissue to extract really good DNA, but it'll be good to see if we could find some of the species here. So if we could just pick one of these bottle brush corals to focus on and then try to zoom in. Oh, that's interesting. Can you move towards the base? Move towards the base. I don't have the quality of video that I did when I was on the ship. I don't have the quality. Do you want to maybe grab that, throw it in the bio box and check out later, see what that is?
Sure, Jeremy, we can do that. Would you like the whole colony from the base?
Yeah, I'm curious to see if it's if that's one stem and then it's got 3 long branches. There's a lot here actually on this one rock. This is a really a lot of high diversity here.
Okay, we can spend some
And
time
of
here and
course
look around.
okay, cool.
Morning to everybody on the YouTube chat. I hope we've got a few fish ID people on there this morning.
Yeah, there's so much here. I see some parentopathes, black corals. Oh yeah. Yeah, this is actually a species we're going to target when we have liquid nitrogen on the boat to snap freeze the tissue. So we won't need to collect that one now, but that's exactly the species we're looking for.
Okay. You want the full colony from the base, right, Jeremy?
Yeah, please.
Good morning, Adele. Any hail in Brisbane today? Okay, we got that one. We'll just have a good look at that.
Great. Yeah, yeah, it's a really interesting colony. It'd be great to see what it looks like. We can take a picture of it in the lab.
Yeah,
Really cool.
we'll zoom in and get a good picture now, and then we'll put it in the bio box.
Oh, that's really interesting. So it's branched and it has pinnules. Seems
Jeremy,
to have
what would you like
multiple
us
rows
to
of
call
pinnules.
this in the event log?
For now, just be safe and call it schizopathid.
Got that, thank you.
And yeah, you could see it at the base. It has the basal attachment, and then it seems like two different colonies have fused. And then the colonies are branched. Yeah, probably one of a species that maybe likes to be hanging on the underside of rocks, because you can kind of see them not standing on the tops of the rocks, but rather out and underneath the ledge. Yep. That's great. That was beautiful. So you can see the pinnules are alternating on either sides, and there's distinct rows of pinnules. Yeah, great specimen. I'm not sure if we collected something like this on the last trip. We'll have to see when we get pictures.
Okay. Put that one in the box for you.
Thanks, Marty. So a little update on the corals that were collected on the last trip. So we collected about 105 corals, and all of the black corals, we've extracted DNA from all the specimens. We've got really good DNA, and a lot of these specimens, you know, they're deep, and rarely are they collected because you need an ROV to view the coral and then to actively collect it. They rarely do well when they come up the dredge. So not many specimens like these have been collected. And when they are collected, they're often given to a museum, and maybe 20 to 30 years later, someone comes along and tries to extract DNA. And at that point, the DNA is very degraded and hard to work with. So we got fresh tissue samples, and we now have fresh DNA, so we could sequence the DNA and understand why the black corals, at least, why they look so different from one another, even closely related species. So we understand how the different species have evolved. And we're providing a checklist of black coral species in the Coral Sea, which is also happening for the first time. And we also collected soft corals and sponges and hard corals. And all of the sponges that we collected are undescribed species, which is really cool. We didn't collect that many sponges, so we're going to also try to focus on collecting sponges. In particular, the carnivorous sponges, so sponges that actively eat little microscopic animals out of the water. So to do that, we're going to need to look really closely at the rocks because they're often only a few millimeters in height. So exactly where we are now, we can look at the rock zoomed in, and if you're able to see little, I mean, they don't really look like sponges. They kind of look like almost bryozoans, I guess. But if we're able to spot some and collect them, they're probably going to be new species also. So that's really cool.
Well, Jeremy, you know how much we like to zoom in and look closely at the rocks, so that will not be a problem.
Yeah. Awesome. Yeah, absolutely. Twofold. Are you also collecting some rocks on this dive?
We'll wait and see what we see. We're really going to prioritize the sampling for Tom and yourself on this dive to make sure that we allow plenty of time for that. But we'll see what we see once we get up in towards the well, shallower part of the dive.
Okay. So yeah, let's look closely at the rocks here. Maybe try to see something small. I'm going to save most of the collecting of the corals that we're seeing here for when we have the liquid nitrogen on board, so we could properly preserve the tissue. There's a fish I see. What are they calling it? A pinecone fish?
Pinecone fish, I think.
Cool. You could see some little azendelic cup corals. Those are the deep sea scleractinian corals, those orange. And we see a bit more of what we've collected.
So Jeremy, it sounds like you've been putting in some long hours in the lab since we got home from the last expedition.
Oh yeah. So my wife and I have been extracting DNA for 3 weeks. So yeah, it's been a lot of work. And even though we're collecting fresh tissue samples, just the fact that they come from the deep, where the temperature is, I don't know, what is this? How deep? Oh, we're not that deep, so it's pretty warm.
Yeah, it's
but
18
when
degrees.
we're collecting from 2,000 meters, where the temperature, yeah, so it's not that cold. But when we were down at 3 degrees, and then we're coming up shallow, the difference in temperature, the corals weren't used to that warm temperature, so the DNA started to degrade even while in the water. So for certain specimens that were very deep and collected at the beginning of dives, we had to extract DNA like 3 or 4 times from a given colony just to get enough DNA to sequence. But luckily, we did collect a lot of tissue, so that wasn't a problem. Yeah, there's a few species here. It's really cool. If we can look closely at this rock here, there seems, maybe just see if there's anything little that's hiding in the crevices of these rocks. I'll try to full screen. I don't know if you remember, Marty, on the last expedition, we collected a carnivorous sponge that it looked like a sun, like it had a circular center and then like radial like appendages, I guess. Do you remember that one?
Yes, I do remember that one. Had us stumped for a while as to actually what it was.
Yeah. So Merrick Ekins at the Queensland Museum is super excited about that and the other carnivorous sponges that we collected. And he said that he often consumes the entire colony when describing it because you need to use the scanning electron microscope to take an image, which means coating it in gold and then shooting electrons at it. So afterwards, the sponge is no longer a sponge that it was when it was underwater. So it's important to get a few colonies, one to use to describe the species and another to preserve it how it looked when it was in the water, so we can later compare other specimens to it. So if we could find some more of those, that'd be cool.
Okay, that's interesting.
Yeah, it's hard for me to see the details close up.
It was quite deep that we saw that particular one from memory.
Yeah, we're only at 235 meters here, so.
Jeremy, do you want us to continue having a look on this bommy, sort of zoomed out a bit and have a bit more of a broader look around, or should we?
Yeah, let's look around.
Yeah.
Yeah, we can move on. So one thing that we never found in the entire month we were at sea in the deep was a whip black coral that had polyps that go all around the colony. So the genus Ceropathes. What we did find was a lot of Stichopathes. So as we're doing this exploratory dive, it'll be really good to look closely at those whip black corals and try to see if any of the colonies have multiple rows of polyps that go around the stem, which would be like this one we're passing right now. Like if you could just look and see if, it has multiple rows of polyps. So for some reason, we couldn't find any of them on the last dive. I couldn't explain to you why. Yeah, they're both extremely common genera in the shallow and in the deep. So it's weird. So if we can look, obviously when you stabilize and then look closely, it might be hard for me to see, but if you can detect polyps that are going all around the stem, it could even. From
what we can see at the moment, Jeremy, it does look like that is the case.
Does it have 8 tentacles or 6? Does it have 8 tentacles or 6 tentacles?
We're not quite zoomed in enough yet to be able to say. We'll just stabilize here for a little minute, and then we'll zoom in and see if we can get a good close-up look. And good morning to Robin Beeman, onshore in Cairns. Rob is the chief scientist on this expedition, and at the moment he's joining us remotely. But in a couple of days, we'll be making our way up to Cairns, where we'll be picking up 3 more scientists, including Rob. And then we'll continue on our way northwards to the far northern reaches of the very outer edge of the Great Barrier Reef.
Yeah, while you're trying to get a zoomed in view of this whip coral, which could be a soft coral, I'm going to have to refer to you. I know you've seen enough of the corals I was working with. I think you can.
Jeremy?
All right,
we're counting,
so then,
so the polyps are all around, but there's 8 tentacles.
Totally. So it's a soft coral, a whip soft coral. So certainly we collected a few of these, so we don't need to collect any more. What's interesting about these unbranched colonies, so soft corals and hard corals are actually very distant cousins. Black corals are more closely related to hard corals, which look much more different to black corals than soft corals do. The reason why they look so similar is convergent evolution has kind of forced the morphology of black corals and soft corals to look similar because they live in similar environments. So this is the optimal branching structure for it to obtain nutrients. So it's actually very interesting to see how black corals and soft corals have evolved. They've evolved very similarly over time, despite them being not too closely related. There's actually really interesting work going on with Andrea Quattrini, who is the curator of corals at the Smithsonian Institute, and she recently discovered that about 250 million years ago, when there was an extinction event and all of the hard corals in the shallows went extinct. So what we know now as hard corals are fairly new. There was an old type of hard coral that went essentially extinct. And what it did was it opened up this environment, this niche became available. So there was all this shallow habitat, but it was unsuitable environment for hard corals because they're made of calcium carbonate. So the acidic level was too high for the calcium to be made, and therefore corals that don't matter how the pH is, were able to exploit that niche. So what happened was the soft corals and the black corals exploited that opening and took over the shallows. And so now today we see lots of soft corals and black corals in the shallows. And when you look at a phylogenetic tree, so it depicts the evolutionary history of the species, you can even see that exactly, or around 250 million years ago, there was this explosion of shallow water species for soft coral black corals. So this hypothesis of this extinction event that occurred 250 million years ago fits perfectly with what we're seeing now that we're sequencing these the DNA and piecing together how they evolved. So I'm just reading the comments on YouTube, and my partner and I, we've been extracting DNA for weeks now. It's a very nice couples activity, but of course she's the boss when we're extracting. She's very systematic and careful, and I can be a bit messy, so she takes the main role when we're doing our extractions.
We're having a little chuckle in here, Jeremy.
Oh, and why is that?
No comment. Put it this way, I don't think there's been any popcorn spilled in the library yet on this trip.
All right. Look, no one can prove that it was me that was dropping all the popcorn kernels in the carpet. Everyone was eating the popcorn. So you can't say definitively that it was me. So I want to say hi to everybody, all the constant viewers. I know you guys are as glad as I am to have Falkor back in the water. There are some substitutes, some other expeditions going on, but Falkor does a really good job at allowing people to
we've just got a
communicate and ask questions. Oh, cool. So yeah, thanks for Falkor for giving us this platform to communicate and share our research. I see a fish, the trevally.
So we're just moving across a bit of soft sediment now at the bottom of the slope here. You can see it's quite biotubated.
It would be good to actually, what would be really cool is if we can get down to the sand level and look to see. So there's a black coral that we collected, I think 2 of. So they're in the genus Bathypathes, and what's weird about this particular species is that it's way shallower than the deep Schizopathes. So this is a family that occurs between, 1,500 meters and about 5,000 meters deep. But there's one species that looks just like them, but is found between 200 and 300 meters. So it's really weird. Preliminary molecular work shows that it's in the same family, but different from the rest of the genera in that family. But we are using a novel molecular tool that hasn't been used before. So we could be, we're in a better position to understand how it fits in with the rest of the species. So it's really thin branched, small. It looks almost like a weed. It has one stem and it has 2 rows of pinnules or repeated branches. They're really small and, so they're hard to see, but I don't know, Marty, do you recall that kind of, that specimen we collected a few of? I think I pointed it out, and I could send you a picture later on for future.
Yeah, maybe send us through a picture.
So,
it's all a blur for me, Jeremy.
Yeah. Yeah. Me too.
I do remember some specific
What's
highlights,
really
so.
cool about this particular, so it only really occurs in the sands. So when we're at this depth, it'll be good to look at the sand, but also at the rocks. So what was really cool about this one species is that, so black corals have skeletal spines, these less than 1 meter tall spines that occur along the entire skeletal structure of each colony. And they're very informative in identifying and separating species. But this particular species, this weird shallow water but deep water species, also has very distinct spines on the basal plate. So the part of the coral that attaches to the substrate, it has these really fine and long spines that no other species has had. And this has never been documented in literature. So if you look at any of the papers, no one's ever discussed this weird feature for this specific species, but we found them on our last expedition. So this is brand new information for the black coral field. And now we need to collect a few more of them to try to confirm and describe the different, describe how those spines are situated on different colonies. Can we look closely at this branching one next to these bottle brush ones? So on the left, it's kind of, yeah, down a little bit, to the left a little bit. Yeah, that one. A little more to the left. Up a little bit. So it's at the top of this rock, this balmy head.
Got a little bit of current here today, Jeremy, so the guys are just stabilizing their position.
Yeah, no worries. Okay, yeah. That one, let's see what that is. That's interesting. So there is one family of black corals that we actually never collected. So we collected 6 out of the 7 families of black corals. And it's pretty weird that we didn't find one family. It should be here. So it's Leopathidae.
And is that one of these branched ones?
Probably not, but let's look.
Yeah, absolutely.
So Jason in Michigan said, last expedition, most black corals on the left had worms. Yeah, so those bottle brush colonies tend to host lots of worms, and they're certainly a really big target for starting in a few days. So we have a few marine biologists who are based in Townsville who are waiting for Falkor to get up to cans. And then so Rob Beeman and Valerie and Joan are also going to join. And they're also bringing a dewar of liquid nitrogen, which is going to allow us to preserve tissue way better. It's weird. Looks like something has grown on top of the coral, maybe. Is that correct? Is that what I'm seeing? Yeah, the thickness, I'll just wait for a clear, sharp view, and then you guys can tell me what you're seeing if I can't see it on YouTube.
Okay.
Yeah, the
Do you want us
thickness
to
seems
zoom in a bit
really
more?
inconsistent.
Oh, we're all the way in, so.
Okay, maybe just look up a little bit. I mean, it's hard for me to see it, I guess, but does it look like something has grown on top? I mean, I don't see any. Do you see any tissue, any polyps?
Not really.
Okay, so then yeah, it seems like
maybe on the
something's grown on top of.
It does look like something else is over an existing stem.
Yeah. Okay, that's fine then. We'll leave that alone to be taken over. But then behind it, so if you go past it and up a little bit, there was another branching coral that seemed more in spirits, a bit more. Not the bottle brush, the one behind. Yeah, that one. What's that? Is it possible to get closer to be able to zoom in more? Or is the current is it challenging with the current?
It is a little challenging, but we'll do our best.
Okay, it seems like on the main stem and branches that same thing has been growing over the colony, but there's obviously something, the top bit's alive. So just curious to see how many tentacles are in one polyp.
They are tiny, absolutely tiny. I don't know that we're going to be able to get in close enough to count tentacles.
Okay. Yeah, it may be a soft coral anyways. Not that I don't love soft corals, but where is it? That's interesting. Let's look for another one like this that might be in a little bit better condition and then grab it.
Yeah, we can just look around for a little bit. It looks like there's something behind it, further behind it. Can you see that just a little further behind the one we're looking at now?
Yeah, let's check it out. Yeah, it's a great location, really healthy, a lot of diversity, a lot of different species we are certainly interested in. I know Tom is very interested in this site as well. He had worked on this reef when he was doing his PhD. So he's really keen when we get a bit shallower to collect some hard corals, some aquaporas. A bit challenging to find. He's looking for a specimen, a species that's kind of cryptic, so it's really hard to see. Marty, I think, right, he talked to you about it as well?
Yeah, for sure. We chatted about that one, and I've had a look at a few images. Hopefully we can find that one. I know Tom's been looking for it for a very long time. I think he's been coming here for at least a decade or more. And so yeah, this is a really high priority dive for Dr. Tom Bridge from the Museum of Tropical Queensland. He's the coral curator. And I know he's been trying to get vision and samples from down here for a very long time. So the last couple of days we haven't been able to put the ROV in the water. The conditions just haven't been conducive. We've got some pretty strong currents out here. And so today we're very lucky to be able to get the ROV down on this very high priority target site. So we're looking forward to a really good dive today.
Yeah, what's straight ahead? Yeah, we're zooming in on it. So, it's I would almost say it's like a, I mean, it's really big. Could it almost be a bryozoan?
Not sure. It doesn't immediately look bryozoan-esque.
Yeah, I mean, not bryozoan. All right. Why don't we grab a branch? A branch of this colony would be fine. It seems like one of those outer branches, like to the left or to the right, seems pretty healthy.
Yeah,
And
okay, we can do that.
Okay. And you can let me know what it is after you've taken pictures of it on the ship. It's a shame that I can't be on the boat. What are, another opportunity to spend a month at sea and keep collecting. I mean, this is
we missed you when we were snipping jelly tentacles the other day.
It's now, well, that's one of the few opportunities I'm happy to not have to go in. Those are challenging. How many did you collect on the last side?
We collected a full complement of 7 jellies on the previous dive, and Luke and Vicky were all over it in the lab, snipping tentacles and taking photos. They did a great job.
Awesome.
Jeremy, do you want us to snip this top section here? Take that?
Yeah, maybe like 12 inches, something like that. And potentially the pink thing next to it, do we think that's a stylastrid? I'm not an expert in things, and I'm not an expert in anything, but certainly not these water hard corals. Well, they're not hard corals. These are lace corals, the stylastrids. Yeah, if you want to just snap the top, I guess that would be fine. Whatever you get is enough. Yeah, Carolyn says the coral reminds her of blood vessels. Certainly looks vascular. Yeah, I wouldn't know what to call this coral. It's really interesting.
We can call it the interesting mystery.
There you go. You could put that on Squiddle. Nice. Okay, got the whole thing. Awesome. Or a large part of it. Well, it's actually good to have the attachment point. That could often be a giveaway to what it is. Trying to decide which bio box to put it in.
Decision made.
Yeah. Cool. Anyone on the comments? Anyone on YouTube or Facebook have any idea what this is? Soft coral? Who knows? Cool. Were there any whip corals on this, Bami?
Don't recall that we saw any, but we can have a quick look around before we move on.
Yeah. Okay. And if we could zoom along across the sand, that'd be cool.
Yep, we'll do. We're just sorting out our position relative to the ship at the moment, and then we'll carry on.
If I send you a picture, could you put it on display on the YouTube and Facebook display?
We can try.
Okay. I'll show you what we're looking for, and we can all look for it together as one big community. Okay, I'm sending you 2 pictures.
I can see that you've sent those, and they are coming in, but we're a little low on bandwidth, as you can imagine.
Ah, right. Okay, well, it's there. So, there's a whip coral up top. And some nice sandy habitat. Yeah, so even when it looks like there's not much, when you zoom in on any particular area, you find lots of stuff, which is always very exciting for taxonomists. It's pretty sturdy and stiff, which would mean it could be a black coral, but let's, when we get a zoom in on the polyps, we could see. I know the current's a bit tough. And there's more of those branching corals beside it. Those curious corals. So who do we have in the ROV driver's seat?
In the pilot seat at the moment, we have Cody and Chris as co-pilot.
Hello, Cody and Chris.
Everyone says hello.
So we're looking for it looks like there's one row of polyps, I think, but we'll look closer. So we're trying to find a whip coral that has polyps all around the colony, which would be a new genus for us in the deep here. Or I guess this isn't really the deep, this is
it's all relative, isn't it?
Yeah, for me this is shallow.
Yeah.
For Tom, this is deep. For you geologists, it's all fair game, isn't it?
Yeah, pretty much. It's not showing us its tentacles. The polyps are all around, but we don't have any tentacles.
Yeah, can you go up to the top? Maybe the top of the colony might have some activity, or it could be wrapped up by something else. So the coral could be taken over by zoanthids potentially. Actually, it probably looks like that's the case. Yeah. I learned a lot about zoanthids on the last trip. Still get pretty confused when trying to determine whether something has been taken over by zoanthids or if that's the actual colony. But in this case,
we saw a lot of that, didn't we?
Yeah, it really makes you question what you know. But in this case, the bumps seem really irregular, which would suggest that it's probably something like a zoanthid that took over the coral. So it looks like it was a black coral because it's so stiff. But yeah, I would say it's taken over by a zoanthid. So, or you know, a colony of zoanthids.
Yeah. Okay.
So below the colony, I think to the right, or if you've been spinning around somewhere nearby, is another one of those branching corals, those weird corals. Maybe just take a quick look to see if it's maybe in a better condition. And maybe a view of the fish for 98% of the people on YouTube and Facebook. Oh yes, interesting, isn't it? Maybe it's a golden coral. Now that we can see more of the skeleton, it looks like a golden coral, which is a soft coral. And then, yeah, there's something black growing around it in certain places. But we did collect a sample, and from that, we're going to be able to figure out what it is that we collected.
Okay, so we'll zoom out and continue on our way. Keep our eyes peeled for small sponges and wet black corals.
And small branching black corals. And of course, big, everything. We're looking for everything.
Just bear with us a minute here, Jeremy. We're just sorting out our position. We're moving around a bit, and so is Falkor. So we're just going to take a moment to sort ourselves out here.
Sure. Are you in a room that is ideal for ship movement?
You know the room that we're in.
A little bumpy at night? Do you have to strap yourself in with
cabin.
a
I thought you meant the room right now. Yeah, no, I'm in a very comfortable cabin midships. So all good for me. I'm getting a good night's sleep. Good. Yep.
Nice to see the control room. I'm not sure if I could hear the people in the control room or if you could hear me, but we'll continue to explore the sandy flat.
We're going to have a look at the sandy flat and keep our eyes peeled for that black coral that we saw on the previous expedition. But I think it was a bit deeper, was it?
Well, the one I just showed you a picture of, it was always found between 200 and 300 meters. So this is like the perfect environment
so
for
this
it.
is ideal.
Ideal, couldn't be more. And it has really, really small, super small polyps. And so it's, of course, really important to preserve the tissue once we get it back on the ship. But I know I'm in good hands with you.
Hey Kerry, it's Vicky, just saying hi. Hope you're having fun on the Gold Coast watching.
Hello to all of our friends and family and colleagues on shore. It is Monday, but it's a public holiday in Queensland today, so hopefully a few people are sitting at home, relaxing and enjoying the dive with us. So what you're seeing on the inset on your screen right now is the 3D imagery of the dive site where we're at today. And we're coming up this undersea landslide where part of the front of the reef face at some point has collapsed and come away, and it's caused this big debris field. And it's a pretty big feature. It goes all the way out, I don't know, maybe 20 kilometres out towards the deep sea from where we are now. But where we've positioned ourselves today is just at the bottom of the scarp in front of the reef front. And we're making our way across some soft sediment on the seafloor at the moment, and then we should come to that steep reef slope, reef face, and then we're going to climb our way up there towards the through the mesophotic zone, the lower mesophotic to the upper mesophotic zone. So the mesophotic is the twilight zone, so it does get some sunlight. So we will definitely have corals there, and this is quite a specialised area of study, quite a different habitat to what you see in the shallow coral reef up on the surface at sea level. So different species and different communities coexisting together there, and it's a place that's not easy to get to, not easy to get vision, and that's why we're here.
Yeah, and even being here, it's hard to see some of the smaller things that are living. Would it be possible, if the current's too strong, feel free to say no, to settle down on the sand and zoom in close to see what small things are living in this area?
Yep, absolutely. We can definitely do that. We are happy to sit down and take a little pause for a moment.
Awesome. Yeah. Sometimes in the last trip, sometimes we would have to stop, in order for the ship to catch up to us or whatever. And only then did we find these really small things living in these seemingly empty habitats. So.
Yeah, for sure.
Oh, what's that?
Yeah, we just said the same thing at the same time.
See,
Something
how
that's
could you
broken
ever
off
see
and
this if
blowing
you're
around.
driving your... Yeah. Ah. Oh, cool. Maybe a soft coral. If you could look eye level at like the seafloor, so like looking out along across the seafloor, you could often see little things that are poking their heads
Sticking up.
at like the horizon, if that makes sense.
Yeah.
If you wouldn't mind just turning the ROV upside down so the camera's closer to the ground, could you do that?
We're good, Jeremy, but we're not that good.
So like, I guess looking down a little bit now to look at the, yeah, I don't know, it's hard to say, but
Yeah, we
like rolling the dice
understand.
eventually. You know,
Yep.
yeah.
Looking down a little bit and zoom in a little bit.
Hmm. Ooh, what's that something? I feel like Dougal now, you know, oh, what's that?
We had a great dive for Dougal the other day. It was good.
Yeah, I saw. Yeah, that's something. Those are some things. So yeah, I'm not sure. Are we far away from there?
They're actually quite far away from us right now, so it's a bit hard for us to get them in focus. But we'll just see how we go here. We can move in a little bit. Yeah. Yeah.
Hmm. Yeah, not sure what that is, but it's something.
Yeah. Sorry, Jeremy, just bear with us a little bit. We're just sorting out. We're all moving around a bit here. Falkor's moving around and Sebastian's moving around. So it's just a little bit tricky.
Whereas I've got all day. So you could see with the inset, so those are shadows of rocks. Is that correct?
Yeah, that's right. Yep. So we just need to keep moving here for a little bit, Jeremy. So we're just going to pick up and along we go.
You're not worried about that climb.
Interesting bioturbation here. We've got quite a lot of pock marks, divots into the burrows into the sand, and quite a lot of little cones coming out of the sand. So presumably those are made by different creatures that are burrowing there. Could be worms, could be shrimp.
Yeah, I look forward to Rob getting on the ship and trying to figure out what's causing those holes and mounds and patterns. Yep. So someone commented about the excitement of Falkor coming to Australia. So Australia does have the leafy sea dragon, which, yeah, unfortunately, it's only found, I think, in southeast Australia. Certainly not in the right place of Australia to see it. Yeah, I haven't
They're
seen
in
one
the
myself. The
they're in the
water's too
cold water,
cold
aren't
for me.
they? Yeah.
Yeah. I stick to the GBR during, when it's 30 degrees. That's my temperature for diving.
Need to invest in a dry suit. Check out the cold black corals.
That's why we have ROVs, Marty.
You've had it too good for too long, Jeremy.
Well, I mean, look, you keep giving me these opportunities to collect remotely. I mean, I'm never going back in the water again. So there was a question about what we're likely to see here today. So at this depth, where we have rock bommies, we're going to see lots of branching corals, lots of black corals and soft corals, a lot of which we've already seen. And towards the end of the dive today, we're going to move toward the mesophotic. Ideally, if the weather cooperates, we're going to get up to 80 or 60 meters. And so we're going to see really big changes in the composition. We're going to shift from obviously corals that don't rely on sunlight for growth to corals that are photosynthetic. And so we'll see lots of hard corals, zooxanthellate hard corals. And then again, different types of black corals and soft corals. So we're going to see really, really high diversity of hard corals in the shallows. So, oh yeah, good spot.
Thanks, Cody.
Gotcha, Jeremy.
So you could see the polyps are extended away from the branches, which would suggest that it's an octocoral. Can you confirm? Do I see 8 tentacles?
Yes, that is confirmed.
Thank you. It is interesting though. I mean, we are collecting soft corals as well. And honestly, I don't think we saw soft coral like this in the last dive. Maybe it's worth collecting it. Are you okay? Is it all right to collect?
Yes, octocorals are covered. They're okay to collect those today.
Cool. Yeah, why not? How big is it? Do we have scale?
Just give us 1 second. Can you see the lasers, Jeremy, with about 15-ish centimetres, 15, 20 centimetres?
Oh,
Those
okay.
lasers are 10 centimetres apart, the folks at home.
So it's not too big of a colony.
No.
Okay. Let's throw it in one of them bio boxes.
Shall do.
And so we have collaborators that work on the evolutionary history of soft corals. So we have Kathy McFadden. We also have Andrea Quattrini, who focuses on bamboo corals. And there's also an interest to get bamboo corals from the deep. It was an interesting colony.
On our next dive, hopefully tomorrow, Jeremy, we're hoping to revisit the Noggin Canyon that we had to abandon on the previous expedition. And so we'll be starting off at about 900 metres on that dive. if the weather and conditions and currents are favourable, which they are proving very challenging at the moment.
Yeah, all right. Well, that means it'll get better, I think. Yeah, it's interesting because, I mean, soft corals can have a whole suite of different branching patterns, but often when you see these soft corals branching, they have some sort of systematic branching pattern. But this kind of seems random. Maybe it's attributed to the fact that it's kind of in the sands where currents are flopping it back and forth. So, you know, it may be due to the environment that it's branching the way it is. I don't know. Yeah, if you can grab whatever it's attached to, maybe a rock.
Maybe a nautilus shell.
No, they reserve nautilus shells for black corals.
Did you ever manage to make your 3D photogrammetry model of that? Or was it a bit challenging?
I think it's doable if someone has the patience to take the hundreds of images required. It would be really cool to do. I don't know if I'm the kind of person that will ever do it.
Let me guess, that person's not
but it's
you, right?
available for someone to do. Christina, would you like to take some pictures? So that this coral is attached to a rock, is that right?
We're not sure if it's just a lump of mud or if it's something hard.
Okay. All right, cool. Oh, good colony. Thank you, very good.
No problem. We'll just dust ourselves off here and carry on.
Yeah. Yeah, and we certainly want to maximize our chances of collecting in the shallowest possible depths for this dive. So if that means having to, you know, not move so much or you know, spend as much time down in the deep as possible before coming up, just, you know, whatever maximizes our chances of sampling at 80 to 60 meters.
Yeah, I think that's a pretty high priority for everyone in the room at the moment and everyone on shore.
So there's a question about black corals and whether they are a type of soft coral. So black corals and soft corals are different subclasses So black corals are hexacorals, and soft corals are octocorals. So they're fairly distantly related. And they're also made of, so soft corals are made of gargonin, which is a protein, and black corals are made of chitin and scleroproteins. So yeah, they're made of different materials. They have different numbers of tentacles. Black corals have 6 tentacles, and soft corals have 8 tentacles. But despite their differences, they do look fairly similar in the way that they grow. And I mentioned this earlier because of convergent evolution. So in where we're looking right now, you'll find black corals and soft corals. And for them to maximize their efficiency in growth and feeding, they need to grow in a certain way that will maximize their ability to obtain nutrients. So whether that is having very few branches and very thick stem, or being highly complexly branched to maximize coverage in the water column, they will look similar despite being very different, coming from very different origins.
These bioturbation pinnacles are really quite large. I'd love to know who's making those. But unless we see the culprit, we won't really know.
Well, we could always just sit the ROV down for 2 hours and see if something turns up.
Wait for him to come out.
Yeah. Okay, settling down.
All right, Cody's settled in now for 2 hours. We're going to sit here. You said it.
Awesome. Yeah, no problem.
Shall we zoom in on some of these
So
little
these
things?
little, yeah, why not?
What are they?
So these could be bryozoans, maybe. I don't know. We have to look closely. I tend to think everything is a bryozoan if I can't identify it as something. So take my advice with a grain of salt.
I thought we were going with sponge. If we don't know what it is, oh, it's a sponge.
Sure. I mean, even sponges and
These
bryozoans
don't look very
can be
spongy.
I can confuse them too. No. But they would look. This would be the size of coniferous sponges. Like the one, if we could zoom in on any one of them, like really closely, and then I don't know, maybe. Actually, is it time to do a contrast, a white balance? I don't know.
No, we're good for now.
All right. Good. So. Yeah, maybe hydroids and GC
I have no idea what
suggested.
these are. See if we can zoom in on
Yeah,
something
maybe.
that's a little bit closer. What is this guy? Maybe hydroids.
Some people are calling it unremarkable, which is kind of rude.
Different strokes for different folks.
Well, I mean, we could all agree that rocks are fairly unremarkable, so at least we all have that in common.
If I recall,
Yeah,
some
I
of your
don't see
best
much
samples
here.
were attached to rocks. Got a little shrimp right there.
Yeah.
Yeah.
Well, is that Is that not what makes it remarkable? The fact that it's, you know, a base for something remarkable.
The rocks are the foundation of the ecosystem, Jeremy.
Sure. And Alameda is the foundation of that, is it not?
Well, hopefully we might see plenty this afternoon once we get up into the, yeah, maybe around 70 metres. We'll start keeping our eye out for the Alameda debris at the base of the reef front. We'll see the debris before we see the living Alameda.
Any idea how fast the current is?
Yeah, it's a little over a knot at the moment and kind of, yeah, changing direction at times. So it's blowing us around a little bit. Quite challenging for Falkor to maintain its heading and position on the surface at the moment too. So the guys are kind of juggling all of that at the moment.
Yeah. Is there a chance that the dive might get called early if the conditions worsen, or does it seem pretty stable?
Anything could happen. Too early to say.
Anything can happen. Yeah, okay. I just know certain people would be disappointed if we didn't get up to the shallows. So if, you know, if it seems like the weather might not fare well, maybe we can get shallower a little bit sooner. But we could play it by ear. So there's shrimp in there?
Yeah, it was a cute little shrimp in there. Might have been two, I think. Yep. Not a shrimp party, but still pretty cute.
No, not. I mean, we don't know what's in the hole. Could be a whole cave of shrimps partying. So
we're being encouraged to go noodling for shrimp.
So there are small rocks around. It might be worth zooming in quickly on some of those rocks to see if anything's growing on them.
We're just doing a pilot shift change here, Jeremy. Just bear with us a minute. I think you might just be seeing a picture now of our 3D seafloor image. And that's ROV SuBastian, obviously not to scale.
So there's a question on Facebook, which I just now opened, about if we're seeing damage with bleaching at this depth. And the answer is no, we're not seeing bleaching down at these depths, which is always a good sign. So I don't think we saw any bleaching in the month in our last expedition. In the shallows, of course. I mean, I'm sorry, in the deep. But when you get up to about 50 meters, you'll start seeing evidence of bleaching.
We're going to sort of fly over this sand, soft sediment area here. Gosh, there's lots of bioturbation, isn't there? Those divots are huge. We're going to try and make our way to the base of the reef front, and then we'll start our climb. But we'll keep our eyes peeled for anything that looks like it's sticking up out of this sand.
Sure. Yeah, if you just periodically zoom in on some of the rocks, that would be fine.
Yeah, oh, little fish. Yeah, so we're not actually seeing too much in the way of rocks, Jeremy. If you've got some little dark patches on your screen, what we're zooming in on now, they look like they're just bits of, well, this looks like a bit of old sponge. Yeah. We'll have a little look around. Thanks for joining us today, Jeremy. What would you have otherwise been doing on your public holiday long weekend Monday?
Oh, just preparing for tomorrow's molecular work. So nothing. Yeah, we're nearly done extracting, and we're going to sequence all of the DNA that we collected, well, from the specimens that we collected on the last expedition. So we should have results back by the end of this year, which is exciting. So we could have a phylogenetic tree of all the deep sea species that we've collected, including the shallow water species.
If you've just recently joined us, we're off the outside edge of the Great Barrier Reef in the central part of the Great Barrier Reef. We're about a little northeast of Townsville, about 65 nautical miles offshore from Townsville, and we're on the outside edge of the Great Barrier Reef. And over the next few weeks, we'll be making our way further and further north. Our plan is to head all the way up the outside edge of Cape York Peninsula, where it's very remote, and it would be an understatement to say underexplored on the outside edge. There's a lot of places in the Great Barrier Reef in the shallow parts that are quite well studied, and we know a lot about those shallower parts of the Great Barrier Reef. They're well mapped and well studied by scientists, and marine park managers periodically visit those reefs for long-term monitoring. but the really remote outer reaches, especially on the outside edge of the Great Barrier Reef, where it's deeper, it's very difficult to get to. These areas are really underexplored, and we've never had the opportunity to have a science ROV in these places
to sequence complete genomes of corals and their hosts, invertebrates living within corals, and also symbionts, so also zooxanthellid and other things that live alongside or within coral colonies. So we're trying to understand how different species that rely on each other have co-evolved. So they were looking for someone to provide them with perfectly preserved deep sea tissues of specific species. And so this trip was just starting, so we quickly got the materials needed to be able to preserve tissue, which would be using liquid nitrogen to snap freeze and preserve it in -80 degrees. And so we've made it happen. So we have the opportunity to collect specimens and completely sequence their entire the entire genomes of these different species, and then compare how they've evolved. And the hypothesis is that they have evolved together. So, you know, when one has changed, the other has also changed in order to accommodate the other. So there's really interesting co-evolution questions that we can answer because of this trip. So it's really exciting for us.
Yes, we hope to have some really interesting dives coming up. Well, they're all interesting. But as you know, once we pick up the rest of our science team from Cairns and make our way northward, and we'll be exploring it, mapping using RV Falkor's sonar multi-beam echo sounder, getting some really high resolution seafloor mapping done. We're particularly interested in the submarine canyons that connect the shallow continental shelf with the deep coral sea basin. So we'll be mapping those, and then we'll be selectively choosing ROV dive sites where we'll put SuBastian down and exploring these features as habitat, and see what different communities are living there. And we're tasked with collecting samples for a variety of different collaborators that we're working with, including Jeremy. There we go. Here's an example on the screen now of what we're talking about when we're talking about our high resolution multi-beam bathymetry. And we can zoom in there, and these are the submarine canyons that we're talking about. It's quite spectacular.
And
Beautiful.
the other amazing thing about having these maps is that we can put together all the specimens that we collected and, you know, put a dot where each one was collected and identify the species. So we have this amazing map and this amazing biological information all together in one place, which rarely occurs. You know, you often see papers that, you know, describe the geomorphology of a place, or you have a paper that, you know, is a checklist of species of a given location. But having those two pieces of information together give you that complete picture of, you know, what's there and how is it distributed and what's allowing biodiversity to flourish, or what's minimizing the ability for, you know, high biodiversity. So that's why this collaboration is great. You know, of course, at times we're going to fight, you geologists and us biologists, but we come together and we're going to put together some amazing papers, and, you know, completely understand the Great Barrier Reef and the Coral Sea.
Absolutely. So what we're looking at now is the front of the reef slope where this undersea landslide has given way. We don't really know exactly when, sometime in the past. This is a really big feature, and the little track well, it's sort of in the middle of that inset now, is the ROV SuBastian dive track. And here we are flying up the debris field of this undersea landslide, with part of the edge of the shelf, the slope, has come away. and we want to get right in at the base of that very steep scarp there, and that's what we're making our way towards at the moment. And then we'll climb vertically up that steep face, and then we'll start coming into the mesophotic zone, and if we get some hard substrate there, we should be starting to see a bunch of different coral communities and algae and lots of invertebrates living in and amongst it, and of course the fishes as well.
Sorry, we're still here. Just taking in the view. Pretty consistent sandy habitat. Not much hard substrate for most invertebrates to settle on to grow. There's a question about what these patchy things are, if it's algae. I don't know. Marty, can you see?
We
said
zoomed
maybe
in
it
and
was
had a little
sponge or-
look. Yeah, we zoomed in and had a little look on one a minute ago, and it looked like it was pieces of sponge. I guess they've broken off and got blown around and tumbled down, and this is where they've ended up.
Okay, we're zooming in. Let's see. Anything sticking out? Oh.
We're getting blown around as well as the sponges.
Huh.
Jeremy, are you seeing just to the bottom left of that sponge waving around. It looks, yeah, looks very soft.
Yeah, looks like a soft coral. Yeah, we'll hold off on collecting more soft corals for the moment unless it's particularly weird. But feel free to take a zoom in to we could still identify it. Not me, someone.
Yep, someone.
Yeah,
Certainly not
like
me
a
either.
soft tree coral. Yeah, it's got that morphology of what I call a soft tree coral, which relates to some genus of soft coral that I can't say. Yeah, an octocoral.
Yeah, might be algae. Looks like it has a tiny little something growing on that.
Yeah. Do you want to waft it and see if, I don't know, would it be interesting to see what's in that pile? Maybe boop it. I know there's some boopers on YouTube that are always keen for a poke.
It's a hermit crab. We'll go check. We'll go say hello to him or her. Wow, pretty eyes.
Oh, awesome.
Wow, he's busy.
Oh, really nice.
He's in a hurry anyway.
Yeah.
Very busy.
Yeah, sifting through the sediment, feeding,
Yeah.
traveling.
There's a hitchhiker on the shell. It's like something that's
Oh, is that an
attached to
anemone?
there. Yeah, I think so.
Yeah. If there's enough anemones, it would serve as protection for the crab.
He's a tough guy.
Yeah. Oh, nice. Looking at you.
I would love to know what he's thinking right now.
Do we zoom in on his face? Or are we zoomed in?
That's as far as we can go. I think he's challenging us to a duel.
I mean, your arm is pretty big, but his arm's also pretty big. I don't know who'd win. Maybe give it a fist bump.
Not today. This guy's all talk and no action.
Yeah, really. First sign. All right, boopers. Here we go. Yeah. Grab a piece and drop it down.
Not a lot going on in there today. Yep. All right.
All right.
We'll head on. We'll keep
call
going.
it- let's call
Let's
it
move on.
algae. Yeah.
We'll leave our little friend in peace.
He's gonna have a great story for his family later. when he goes home. All right, so this crab is called Carl, so everyone's clear. Hello, Jenny, watching from Facebook, I think. So we're just zooming along,
Yeah,
slowly making our way up.
yeah, we might just pause here momentarily. It looks like there might be.
Oh yeah. Oh yeah. Oh, some stuff here. Let's check it out. Okay, so now we have some hard sediment, so we could assume that we'll find some corals attached to it.
Yep, we'll pause here and have a little look around.
All right, so it looks like a soft coral here. Just zoom focus, but looks like a soft coral. Yep.
Yep, I think so.
All right. See what else is in this area. This seems to be living algae, you know, the source of the deep stuff that was discarded, potentially. Lots of little rocks, broken bits of coral, probably from the shallows. I think we could assume there's a really high diversity of hard corals in the shallows above here. There's another octocoral. You can see the polyps are protruding distinctly away from the branches. Can you look behind it, just above it a little bit?
At that tiny little thing sticking up on the top there.
Yeah, Okay, I don't know what that is.
Does that mean you'd like a closer look, or shall we continue looking around?
Now let's continue looking around. There'll be a few things here that are interesting. You can see lots of cup corals, still a zooxanthellae solitary cup corals in the crevices. We collected a whole bunch of them on the last expedition. And those will be looked at by Stephen Cairns at the Smithsonian Institute, and Tom Bridge might also work on them. So soft corals. Nice cup corals.
That's pretty. I don't know what it is, but it's pretty.
Oh yeah. Oh, that's cool. Yeah, let's take a close look at that. Nice color.
We're just going to nudge up a little bit closer so that we can zoom in a bit more.
Yeah, that's nudge worthy. I think people are saying they saw garbage potentially. I didn't see it, but
Yeah, no, I didn't see it either, but we'll keep our eye out for sure.
you know, ROV Falkor is a very environmental robot. He always picks up the garbage that he finds when diving. Well, it's certainly a octocoral.
It's a very pretty color.
Yeah, why don't we grab it? It's actually really interesting. We didn't collect anything like this on the last expedition. So if you're able to.
Yes, okay, we were just momentarily distracted by another thing, but you have our full attention and we're focused on this beautiful blue-coloured octocoral that you would like to collect. And if you look to the right of the pretty blue octocoral, or along to the right edge of the screen, can you see an eyeball in that little crevice there?
I cannot.
We think there might be maybe a tiny little octopus in there.
Oh, cool.
Yeah.
All right.
Oh, that's cute. All right, we'll just figure out our best position to get into to sample that octocoral.
Yeah, it's often hard to collect small corals like these that are kind of hiding in crevices. It's hard to know the scale, the size of this ROV, but it's like the size of a small car. It's like a Volkswagen Beetle. So it's really, really big, and it's got these big claws. So it is hard to carefully collect corals like this. But of course, we have really good ROV pilots that are able to do their best to grab these colonies. Yeah, put arms on your beetle, and it'll look like the ROV. It is October. Is that why we're collecting octocoral or octopus? Okay, octopus. While I have the spotlight, I'd like to share that our new coral genus and species is Coral of the Month on Project Phoenix's website. I just shared it now on YouTube and Facebook. I'm very proud.
Ah, well done. That's exciting.
Yeah, very big
Yeah.
accomplishment. So go check it out. So this is the newest genus and species in the black coral order. That's a small coral, huh?
Yeah, it's pretty small.
The hashtag is EdgeGBR, is that correct?
Yes, that is correct. That's our social media hashtag for this voyage. This expedition is called Northern Depths of the Great Barrier Reef, and our social media hashtag is hashtag EdgeGBR. That is really small.
Yeah, it would be interesting to collect, if possible. I mean, yeah, it's clearly going to be a tough one. If you could just push it, it'll probably break at the base, and then maybe scoop it up. I don't know. Would that be easier? I don't know.
We are getting bounced around quite a bit here at the moment. So
Yeah.
J-Rod and Chris are doing a sterling job trying to keep us
We're in
stable. We are in good hands.
We're in good claws. Oh yeah, it's a bit soft. Maybe sucking it up. Oh well, I'll let you guys obviously know what's best to collect it.
Yeah, we won't try and scrape and suck. No?
Yeah, this is a small soft coral at a depths well beyond what people can scuba dive to see. So given that it's so small, and cryptic, there's probably very little known on this species. So that's the coral attached to, I guess, the algae that it's holding.
No.
To the left. Just let it get
Oh,
a little
yeah.
up. I think that's it. No.
Is that it there?
I'll wait right there, but
This is a very suspenseful moment here at the moment, and we're also just discussing what's for lunch.
Oh, ooh. I do
You know,
miss
it's
lunches
that
on
time.
Falkor.
Looks like we got it. Yep, there it is. Well done.
Oh, wow, awesome.
Yeah, we
That was
found
a
it.
tough one.
Got it.
I'll tell you, I had my doubts. But I shouldn't with this dream team. Yeah, so what we collected seems to be a soft coral, octocoral, really small, brightly colored. So after the dive, Marty is excited to take the coral in the lab and take some nice scales images and subsample for tissue preservation for later DNA extraction. Isn't that right?
We're super excited, Jeremy. I have 2 helpers, and we all can't wait.
Awesome. I'm glad we're all on the same page. So people are asking about bubbles coming out of the lid of the bio box.
Sorry, Jeremy, we're just dancing around here deciding which box it's actually going to end up in. Just give us a moment.
Take the coral in the lab and
Okay, another successful collection.
awesome.
Yeah, I think we'll move on. We'll, onwards and upwards.
Yeah. Is that a yellow thing in the? Is
Right
that
in
a
front,
fish?
in the middle.
Yeah.
Oh, the floaty thing, top left. It's just
Right.
a bit of floaty stuff. I don't think it was a fish.
All right.
I've got all the technical biological terms here. Floaty stuff.
Yeah. Well, look. Yeah. That works. Oh yeah. All right. Lots of stuff going on here. Maybe just
Have
a quick zoom in
quick
on
look
that
there?
branching thing.
Okay.
Yeah, sure. It's interesting. But the thing. Oh. Well, okay. Just another soft coral.
Just another
Sorry.
soft
I thought it
coral.
was something else.
Okay.
Yeah, I thought it was.
Oh, worth a look.
Never
Worth
mind.
a look.
Move on.
Yep. You can see that we're starting to move up the slope now. It's getting quite steep. And all these little blocks are boulders that have tumbled down from up above. Oh, here's a whip. Have a quick look at that.
Oh yeah. Yeah, the way it's kind of wilted, I guess is the best word to use. It might not be a black coral, but we can check it out.
And there's another one to
Often
the left of it as
soft
well.
coral. Let's check it out. Oh, it's pretty stiff.
We look
Yeah,
at this
it's
orange
worth
one
taking a
first.
look. Yeah. Well, unknown explorer is calling it a bamboo. She could be right. Maybe. Yeah, it doesn't look like a black coral. Looks too fleshy for a black coral. So, octocoral. Yeah, we'll hold off on collecting these whip soft corals until we have the liquid nitrogen on board.
Okay. Did you want to zoom in on that one that we were just hovering over, Jeremy, or is that not particularly of interest? In which case we can keep moving on.
The second one, just a quick zoom in, just to double check.
Okay.
Yeah, these guys are tough to focus on. It's a small bit of coral, so you have to focus on what's in the foreground rather than the background. So probably have to manually focus, which is challenging.
Sorry, Jeremy. We're just fiddling with our knobs here.
Yeah, I know. No worries. We all have to fiddle with them sometimes. I'll grab the dragon. Yeah, well, it's nighttime somewhere, right?
Sorry,
Okay,
folks.
so
Just bear with us for a moment here while we sort ourselves out.
there's a question about the bubblegum corals having non-branching morphology, and I would say no. Yeah, I don't think so. Certainly not an expert on soft corals.
Hey, Jeremy, I'm gonna. Whoa, hello.
And I think we lost.
Oh, hopefully we're back.
I'm with you. Yep, we're back.
We're back. Okay. Nice fish.
Oh, there's a fish.
Yeah. We're back. Jeremy, I'm going to step out momentarily and take a short break. I'm going to leave you in the capable hands of J-Rod and Cody. And we've got Luke and Vicky here, and they may hop on and chat with you. And I'll be back in just a little bit.
Right on. So, J-Rod and Cody, how was your time on land? Short but sweet.
Yeah, Jeremy, it was pretty short. Got a bunch of maintenance done.
Yeah, must be nice to have a little bit of time off the ship.
Yeah, it was decent. We didn't get that much time off, you know. It took maybe half a day, if that.
Oh,
Go see
well.
Brisbane.
Hmm.
Yeah, we're gonna do a white balance real quick, Jeremy.
Sure. Yeah, that must mean some light is penetrating down to this depth. We should soon start seeing changes in the coral composition. There's a fish. Plucked Ranthius. Is that a coral there? Probably a soft coral. Okay, another whip. Yeah. The crinoid? I think so. So zooming along at 170 meters. Is the temperature a bit warmer?
Yeah, it's 21.8 degrees Celsius.
Wow. It's what some would call swimming temperature.
Still cold for you though, huh?
Yeah, pretty crazy. If I need to wear a wetsuit, it's a bit cold. So we're starting to see lots of little whips and fish. Maybe someone can identify. Can we look at one of these whips? One of these white whip corals?
Yeah, sure enough. We're gonna get steady, lay down, and we'll zoom in for you.
This is a good opportunity to look at other living things on these rocks, little small things in the crevices. Okay, here we go. Yep, so it's no longer Looks like it's been taken over. Okay.
You want us to keep moving?
Yeah, sure.
Copy.
Yeah, those potentially are zoanthids that took over the colony. Or something else. But yeah, if you guys see anything with your 5K resolution, let me know.
Yeah, Roger.
Hey Jeremy, this is Luke. How are you?
Hey Luke, how's it going?
Good, good. I was just wondering if this is the sort of habitat we get those carnivorous sponges.
I would say no. We were finding them much deeper on the last expedition, but I'm certainly not an expert. So Merrick Ekins, who is watching periodically today, but I think it's just a bit too shallow. But on deeper dives, we'll certainly be looking out for them. But we will have to confirm with Merrick the depth range of these carnivorous sponges. We certainly saw some glass sponges at the beginning of this dive, I think. But yeah, we're getting a bit shallow now. So we could soon start seeing maybe some hard corals in a little bit. So on the last expedition, we collected the deepest zooxanthellae hard corals on the Coral Sea GBR, which was exciting. And that's now at the Queensland Museum. But always looking to beat records. So we could look for another, more deep hard corals on these dives. What's this we have here?
Is it a little feather star?
So this is like a flabellate colony, this pink coral. It actually might be worth getting a view. Yeah, if we could zoom in. Looking at the base and the stem, it's probably a soft coral. That's nice. That's actually probably something we could be collecting after we have our liquid nitrogen. So we'll leave it alone for now. And although this is not the species of coral that you find pygmy seahorses, we did find pygmy seahorse on the last expedition. So when we come across those fan corals, we could definitely take a look.
So it looks like we just saw a little gastropod or mollusk of some description on there, Jeremy.
Yeah, it looked like some sort of gastropod. Very cool.
Jeremy, I should probably just introduce myself. So I'm Dr. Luke Noda from Queensland University of Technology, and I'm a marine geoscientist as well, so similar to Marty. I probably have a foot in both camps, so this competition about geologists and biologists doesn't really mesh with me. But yeah, I'm just filling in whilst Marty's having some lunch.
Awesome. Well, Rob, I'd say Rob has a foot in both camps. He probably is better at identifying the random critter that's collected on these deep expeditions than I am, because he's done so many of them. So yeah, it's always great to see people who are doing both things, you know, seeing the full picture.
So some of the things that I lecture on at QUT is paleontology. So we have to learn our phyla and our identifying characteristics, and how many polyps and how many tentacles and all of those sorts of things. Bryozoans are
Also,
always,
you know,
bryozoans are always in
tough,
that too hard category.
Yeah. You know, to date, a fossil of a black coral has never been found, despite us knowing that they've been around for 500 million years. So I think part of the reason why a fossil's never been found is because so few people know what to look for when they're looking at fossils. So it's my hope that in the next year or two, you know, now we're working on black corals, that maybe someone looks at a black coral, then goes, wait, I think I've seen something like this before, and, you know, finds this really old fossil to help us date the order.
That's actually pretty amazing. We never found a fossil of it.
Yeah, well, the Chinese, so a number of years ago, a group of Chinese scientists wrote a paper about a fossil black coral, but looking at the pictures, it's clearly not a black coral. So yet to date, really, there's been no verified fossil of a black coral, which, yeah, it's odd. You know, they're made of chitin. It's a hard material. You would assume that there must be fossils out there. So I think it's a matter of just looking for them with the trained eye.
So the substrate's changing a little bit as we go up. There's a few of those big Cycloclypeus foraminiferas coming in, which is a good sign.
Yep, we're getting up there. So we should soon see some more life.
They're now my favorite foram, Cycloclypeus.
Do you want to collect any, or has it been done so many times?
Marty got a few on the last voyage, so I'll leave it at that.
Okay. Looks like something up ahead. I don't know what that white thing is sticking out.
A sponge, perhaps?
Sponge? Yeah. Yeah, okay. A whip coral. Yeah, nice looking sponge. Not the particular target for Merrick Ekin's work, but suitable for white balance for sure. All right, seeing lots of whip corals pass by. Can we get an inset of the map, the 3D map, to see how close we are to the top? Oh, it's. Can we look at this? This might be an interesting sponge. So. Okay, here we are. Okay, fairly close. And then pretty soon we're going to see a big increase in the angle. So, okay, this is like a soft coral. Okay, this is like a soft tree coral. Usually you could see little crabs living on it, little shrimps. And there's bryozoans at the base, I think. Those are bryozoans.
Let's just say yes.
I have a colleague at the I have a colleague at the Museum of Tropical Queensland, Robin Cummings, who works on bryozoans. And after each one of these expeditions, we always, we find little bryozoans connected to the bases of all the corals that we're collecting. And most of what we give her are things that she's never seen before. I mean, rarely does anyone ever target bryozoans. And in the deep, they're mostly unknown. So besides our targets, we're discovering a lot of new species of things associated with the things that we're looking for, which is pretty cool. A fish?
A fish. That's as far as I go with that. But there's
Yeah, all right.
some of those cycloclypeus in the front of the view, those grey discs, the benthic foraminifers. I think we're going to sample a couple of those.
Oh, awesome. Yeah, we have our fish expert Kai, who's helping us identify the different fish species that we view. Of course, we don't collect any fish. There just weren't enough people interested in fish when this expedition was being designed for permits to be written for us to allow to collect for fish, but it's always really fun watching Falkor collect fish. So on the Ningaloo expedition, so in West Houghton, there was the claw, the ROV arm had a net, and it was trying to catch fish in the net. So it's like a little kid trying to catch butterflies. So it's a fun thing to watch. But on this trip, all we could do was watch and call it a fish and move on. But that's not to say we didn't make any big discoveries. So on the last expedition, we found that walking fish, who's classification escapes me, but it's rarely seen, and we saw it. We got really nice footage of it on the last expedition. All right, so here's a really large benthic foram. So Luke, is there any reason why these forams are so much larger than the average foram?
So the thing with forams is they're a protist, so they're a single-celled organism. And that's why most of them you have a whole range of foraminiferas, from planktic ones, which are really quite tiny and they float in the ocean, and then there's a lot that are like a sand size that are up on the shallows of the reef and probably down in this depth as well. And then you get some ones like Cycloclypeus that have a big surface area. And they potentially have a symbiont that helps them gain nutrient through photosynthesis, and that bigger surface area gives them an advantage to get a larger area to absorb light. So that's why they specialize in these sorts of depths.
Do we know much about the symbiont?
No, we don't, and that's one of the reasons we can collect them.
Awesome. Well, I only ask because since we're sequencing full genomes of corals and symbionts, we might want to snap freeze one of these to maybe try to extract DNA and sequence from that as well. Maybe we could try.
Absolutely. So you can see they're quite fragile, difficult to pick up, but we'll pop these in some ethanol and hopefully come across some more later on when you're on board.
Yep. So we have Joan and Valerie who are going to be in charge of the liquid nitrogen. And so I just met with them the other day. They're ready to go. They're excited. Yeah. Looking forward to snap freezing some tissue. Really big opportunity.
Snap freezing, anything is fun. Liquid nitrogen.
Yes, it is. Yeah, on the YouTube feed, they're talking about all the fish that we found on the last expedition. So besides the drunken fish, there was also the giant grumpy fish, and of course, I'm using their technical names. Looks like we're getting some algae with it. Yeah, Rob Beeman is keen to, of course, check out the algae. Okay, so we're at 130 meters. Very soon we should start seeing some hard corals, probably some plate hard corals. Probably Leptocereus is the first sclerotinians we'll see that are usually found in low light environments. On the last trip, we collected a whole bunch of Leptocereus, and Tom Bridge is working on those. And he'll probably want a few more. It's always challenging because the ends of these dives are usually when we enter the depths at which people like Tom Bridge are interested in collecting. So we tend to undersample in the mesophotic and the shallow mesophotic habitats, which is why this dive is of particular interest to Tom, because he knows for sure there are hard corals that are present in this reef. So if the weather allows, we'll try to grab some aquapora that don't really look like the normal hard corals. They're not plate-like. They're kind of bramble. Usually found unattached to the substrate, which is interesting. So we'll keep an eye out for those, and hopefully everyone gets to see them. There's a question about the depth of future dives. I don't think we're going to be doing any dives over 2,000 meters. Luke, do you know, or anyone in the control room?
No, we won't be doing dives deeper than 2,000 on this one, on this voyage.
Looks like another soft coral. A whole bunch of corals on this bombing, this little rock. We can quickly look at just some of the individual colonies, and also maybe on the underside, if possible, if the currents aren't too strong.
Hey, Jeremy. Sebastian is working very hard at the moment and getting quite warm, so we're just going to sit here for a little bit and cool down a little,
right.
Fighting those currents.
Yeah, this is a great spot.
But this is a great spot to pause and look around. There's plenty going on here.
Yeah, absolutely. We don't want to overheat Sebastian. How was lunch? Was it good?
I was going to ask you what you were having for lunch at your place today, because I've just had the best fettuccine carbonara I think I've ever had in my entire life.
Oh, that's saying something.
Hey Jeremy, is that a little Leptocereus-y looking thing, brown thing that we can see in the center of the screen now?
Yeah, potentially. You could zoom in a little closer. But it probably is. Oh yeah, I see Leptocereus to the left as well. Yeah, okay. So the ROV pilots will be well aware of the challenges of collecting Leptocereus. So let's reserve collecting them for when they're in like a really good position. They're obviously super, super fragile. And
getting pretty warm. Sebastian is getting pretty warm.
Yeah, okay. Yeah, we'll definitely try to cool down the ROV as much.
Luke's just spotted some live halometer.
Oh, nice.
$124 meters. We did see some live halometer a little bit deeper than this out in the Coral Sea on the Queensland Plateau. I think about 140 metres was the deepest that we saw, helimeter. Gosh, there's a lot going on here.
Yeah. Oh, some bryozoans. Yeah, we can see what's on the side of the rock here.
Do you want us to zoom in a little on to the side of this rock here, Jeremy, and have a look at the sort of close-up stuff there, or are you wanting us to zoom out?
Yeah, sure. Yeah, zoom in. We can maybe find something. And it would also give us a little bit of time to cool down.
Yeah, we're doing okay now. We're good. Yep, we're good.
Okay. So if you to like the base of the rock.
Checking out the cryptic overhangs for things that might be hiding in there, yeah?
What's
There we are, we're maybe about halfway up the wall. Looks like we're about to come up and over a little bit of a terrace there, which is interesting.
let's check it out.
That's a nice view, nice oblique view. You can see that whole undersea landslide where it's come away from the edge of the continental slope there, and all that debris has tumbled down slope and out. So we're looking, the perspective we're looking at now is from the top looking down.
a lot of life on it.
Yeah.
So we're seeing mostly soft coral. Actually, can we look at the inside of this crevice, like to the right?
Yep.
Or I guess really anywhere underneath works, but yeah. Okay. Yeah, you can see how much life
Yes,
attaches to a rock
underneath.
when a rock is available. So, these fan corals are potentially interesting. Maybe get a close up of the two different fans that are next to each other. So we're now entering the photic zone where, so we'll see lots of different species that we weren't seeing just a few meters down. So some of these fan corals could be shallow water black corals.
Can you just direct us to which ones you're talking about, Jeremy? Just so we can get
straight ahead, that big
the white
blue
thing
one,
that's
and then the
okay.
yeah, is that white?
Yeah,
okay.
this one.
And the
Yeah.
next to it. Yeah. So
Wow, it's delicate.
actually, all three things right here.
All three. Okay.
Let's see. Okay. So I felt like Dougal again, just saying that. This is in the family Myropathidae. So this is a really nice black coral. Can we grab a piece of it after taking? So it'd be good to take a few different like stills with the scale, and then we could just take a quarter of it.
Yep. Okay. Sure. We can do that.
Awesome. Yeah, a lot of these fan black corals, shallows, but their deeper species are obviously under sampled. So this is a really good opportunity to see, one, if at 15 meters, or if it's a different species, and if it's a different species, how closely related is this to shallow water flabellate black corals? And we were talking about how soft corals and black corals can look very similar. So the same thing can be true about flabellate black corals in the shallows and the deeps, where they can actually be completely different families, but look identical. And you could see it's got some sort of a critter, crustacean.
Yeah, it's got a couple there, and looks like a brittle star as well.
Awesome. This is a great colony. We don't want to miss the opportunity, so we'll collect it here. But then also, if we see another one later in the trip, we could also grab another one, and that later one we would grab and try to also collect some of the associated critters, so some of these crustaceans. But for now, we could just grab the coral.
So we can just
so you can
take
see
a piece
the
from this
polyps
one?
are really small. Yeah, like
a small
6
handful?
to 12 by 6 to 12. Yeah, like a handful. Literally, whatever you grab and pull, whatever comes off will be good.
Okay.
And then when you grab it, if you could bring it close to the camera, we could look at the arrangement of the tentacles, which is always fun. I know that's what people are always asking for in the comments, close-ups of black coral polyps. So, got to give them what they want.
Such a crowd pleaser.
There's a there's a question. This is not the species that I was hopefully in one of these, in this or one of the later dives, they could show the picture of the target that's found at 200 to 300 meters, and it has one single stem, and then yeah, it's different than this one, which is Myropathidae, genus Myropathy is species, who knows? Oh, sorry, is my audio acting up?
It's a little jittery, but it's okay.
It must be because I'm so excited.
You're equally as excited as Dougal, because it was the same with Dougal the other day too. Just trying to shake off a bit of that algae.
Yeah.
Gosh, it's delicate, isn't it? Tiny.
The polyps are really tiny, which is why when it does get, yeah, that's good. When you get it on the deck in the wet lab, to quickly cut a fragment and just throw that in ethanol and 100%. But so is this as much as you could zoom in?
Give us 1 second.
In other words, is that all you got? So this will be a really good opportunity to show you how the 6 tentacles are arranged around the feeding mouth. If we can.
There we go. See how close we can get, but that is really tiny.
Yeah, they're very small. So that's it's typical of this family to have small polyps, small tentacles. But if you could look closely, you could see that there's 3 tentacles on each side of an individual polyp. So at first you might think that you're looking at 3 polyps, but each one of those is 1 tentacle. So each cluster of 6 is 1 polyp.
There we go. That's nice. That's as good as it's going to get, Jeremy.
Okay, that's great. Yeah, so now you can see.
The guys just went out and got their subsea microscope out for you.
Thank you. So yeah, so you could see each polyp has those 6 tentacles, 3 on each side, except 4 of them are closer to the tops and bottoms, and there's 1 on each side. So yeah, great collection. So I call that a Myropathidae, Myropathies, whatever.
Yeah, whatever.
Just don't call it whatever. That'll be confusing.
And was there something else, another fan-like coral that you wanted us to have a look at on that rock as well? When we were first got there, you said there were a couple there that you were interested in.
Yep. So on either side of that middle one that you just grabbed a piece of, we could just quickly look to make sure it's not something different. The color's different, but you know, the color can be different within a single species. So.
Yeah, sure. We just finished dealing with this situation.
Having a little trouble there.
It's like trying to get the last drop out of the tomato sauce bottle, right? There we go. there we go, tucked away safely in box 1B.
Awesome.
Are we on the right target here, Jeremy?
Oh yeah. So, if we could zoom in a little more.
Where do we get that microscope out again?
Yeah, thanks. So the tentacles look a little longer. It's interesting.
Are you contemplating?
Can we do a can we do a side by side of the two corals? Like just zoom out a little bit to get, you know, both corals in the same
I
view.
see what you mean. So the white one
like
that we just
this.
collected, and it's pretty pink
Right.
neighbour. Are you doing a side by side comparison?
That's right. So the pink one looks a bit more complexly branched, and more layered and bigger. Why don't we grab a fragment of the pink one as well?
Okay.
Nice. Yeah, if you can get that whole bit, that'd be great.
Jeremy, turn your notifications off.
But if I turn them off, I may forget to turn them back on, and I may miss a dive. That's great, thanks. Oh, yeah. Beautiful. Awesome, thanks a lot.
No worries.
And I think I'll just take this opportunity to explain why we're collecting and how we decide what size of a colony, you know, how much of the colony to take. And while I'm talking, if you want to move on to the next, to shallower, that's cool.
Okay.
So, well, the diversity of corals in the Great Barrier Reef and the Coral Sea are mostly unknown. But this information is what we use to protect biodiversity, because if you don't know what species live in an area or how far those ranges extend, you don't have that fundamental biodiversity information that you need to make these informed conservation decisions. So before this trip and the trip just before it, we really had no idea what species were living in these deep habitats. And by the way, these deep habitats make up most of the Coral Sea and Great Barrier Reef. So there are marine reserves in place, but they were created without much understanding about what it's protecting. So one of the aims of this expedition is to collect all the different kinds of corals that we're seeing and use that information to make a sort of species checklist. And this checklist will allow us to know what's in this area, how far within and beyond the coral sea these different species extend, and whether they're rare or they're common, where they're distributed, and also the threats towards these different habitats. So there's fishing, there's mining. So using all of that information, we're able to give this data to the decision makers, the people that decide where a marine reserve goes or what rules and regulations are put in place. And they're able to update what they know about the coral sea and the Great Barrier Reef, and have a better chance at conserving overall biodiversity. If we were to just collect a small fragment of each colony, we probably aren't getting enough of the colony to identify to the species level. So, so people often ask, why are you collecting such a large piece of the colony? Well, a lot of species have features that are informative in identifying the species, but those features vary along the colony. So at the top of the colony, it may point to one species, but at the bottom of the colony, certain features might be different. So for certain species, you need the whole colony or a large portion of the colony. And so we're very aware of what we're collecting and how much of each we're collecting. And on the last expedition, we did a really good job collecting all the common species. So on this expedition, we're going to be much more selective about what we collect, because if we've already collected it, there's no reason to collect another one. So yeah, on this expedition, we're just going to fill some species gaps, some things that we thought we'd find but didn't find. If we come across something really weird, we're going to collect it. Coniferous sponges all turned out to be undescribed species, so we're going to try to collect those. And we also are involved with this brand new project. Well, it's not a new project, it's new for us to be involved in this project where we sequence the entire genomes. So there's certain species that are of great interest to sequence the full genome. And by the way, there hasn't been one full genome sequenced of a black coral yet, and only for very few hard coral species. So it's very exciting to have the opportunity to sequence their entire genomes. It'll give us a lot more information about the species, how they're related to other species, and how they've evolved. So yeah, that's my main contribution to this expedition, is to help determine what to collect and how much of it to collect. And then of course, afterwards, back at James Cook University, work on the molecular. So, extract and sequence the DNA and try to piece together that evolutionary history story.
Yeah, it's a really important dimension to the expedition, and we're glad that we're sorry that you weren't able to join us on board, but we're glad that you're able to join us remotely.
Yeah, it's the amazing thing about technology these days. I can sit at home and, watch a live feed, and it's really amazing. Just to be able to, see what you're seeing, although I'm sure the quality is a little bit better by you, but I can't complain, obviously. So we're seeing,
so
I
much
think,
diversity
soft corals.
here, a lot going on.
Yeah. At any time, if you want to zoom in on really random colonies just to see what we're looking at, it's hard to, choose which to look at. So there's a leptoseries in front of us now.
Yeah, it was nice to do a little fly around that bommy and just get an overview of how much colour and diversity there is on there.
Yeah, absolutely. Yeah. No, often I want to zoom in, but sometimes you do need to take a step back and see the big picture, all the other corals that are surrounding what we're looking at. Some more
anything in
leptoseries.
particular? Yeah. Anything in particular that's caught your eye there that we want to go and check out?
I'd say no in this particular location. We can keep, meandering. So someone commented about how they wish the ROV was around during their PhD. I've certainly collected a lot specifically for my, I mean, it's really helped fill in lots of species gaps in my PhD. It's, yeah, it's a great resource. I'm lucky that it's, you know, happening right in my backyard just as I'm doing my PhD. There's a question about those plates. Those are leptoseries hard corals. Those are scleractinians. So those are the deepest dwelling zooxanthellate scleractinian corals. And you'll often find scleractinian corals are very plate-like in the deep, and then they become a bit more branched as you go shallow, and that's because they do rely on sunlight for growth. And so the plate morphology allows it to maximize the light it absorbs. And then as you get shallower, there's more light, therefore less need to be so widespread, because being so widespread also means strong currents would probably rip you off your substrate. There's some fish in the area.
What is that? Is that a sponge? It's a sponge, yeah. Nice little orange sea star in front of it there. That's nice.
Yeah.
Anything weird and wonderful that you don't know what it is, you just call it a sponge.
Well, this is a typical, I would say, sponge. I've only recently learned about carnivorous and glass sponges, but you know, before that, this is all I thought sponges really were. What kind of sponge? I couldn't tell you.
No.
The kind of sponge that you use in a bathroom. That's the kind of sponge. In the shower.
Jeremy, do you recall, I don't really recall seeing so much of this fleshy green algae around at these depths on previous dives on the other previous expedition. There's a lot of this around.
Yeah, no, especially, yeah, even deeper, right? We started seeing a little bit deeper, so there's a lot of it out here. Yeah, I wonder why.
The little orange one, you want to go check it out? Yeah, zoom on this side. Yeah.
So there's a question about how much light we're getting at these depths, because we're now seeing zooxanthellate hard corals. So quite a bit of light. Well, if you turn off your ROV lights, I imagine we would see pretty well, right?
Yeah, we're gonna zoom in and have a little look at this cute little sea star here. And then maybe we'll check out the natural light situation. We're in 120 meters at the moment, 120 meters depth.
That's always fun to do. And there's a question about glass sponges. So yeah, they have a matrix of spicules. So it's silica, pure silica, that makes up these sponges. And they're really quite beautiful. So
we've just switched off Sebastian's lights. So what you're seeing now is the natural light coming through from above. So that's not bad for 120 meters.
No, you can see really well. So clearly, corals can obtain sunlight at these depths. And there's a question about there being coralline algae down below 100 meters.
Yes, we quite regularly see the coralline red algae. That's usually the first. The red algae is usually the first photosynthesizing organism that we start to see as we're coming up. It's usually the deeper, at the deeper before we start seeing the green algae.
So that's our signal that we're entering the lower mesophotic zone when we start seeing that coralline red algae encrusting over the rocks. And yeah, when we start seeing that red, then we know, oh, okay, there's enough sunlight getting through here. Not a lot, but enough. We're just settling down here, and then we'll be on our way. Oh, fish. Nice. Are you frozen? Not sure if you were frozen there on the screen. All right. We'll continue onwards on our... All right.
See a fish.
What is he doing?
Then
Are they those barbel things that they have on the bottom of their underneath that they feel around with?
yeah, it's
Goatfish, there we go.
been called a goat.
Oh, hi, Will. Will, our fish collaborator, I just saw pop up on the YouTube chat there. Sensing for food. Glad you could join us, Will. Have you been on for a little while and I just didn't notice?
Okay, good to have fish people on board or on chat.
Yeah, it's always handy to have a fish person around.
going to take a closer look at this. I think it's a sponge. Yeah. Sponge, yeah. Okay.
Yep.
All right, we'll continue slowly making our way along and up this slope. Saying hello, Jodie Webster, another collaborator, and in fact my PhD co-advisor, who is watching from Sydney.
Making sure you're hard at work, I see.
Yeah, checking up on me. Not sure if the girls are watching, Jodie, but if they are, hi Lily, hi Scarlett. Yeah, this fleshy green algae is just everywhere, isn't it? We really didn't see that on the previous expedition.
Yeah. I imagine it'll go away, giving way for hard corals in the shallow, or
Yeah.
we're going to have one hard coral taxonomist.
Nice little mollusk shell there. Don't know who's at home, if it actually is a mollusk or if it's a hermit crab in under there somewhere. We could give it a little nudge and find out. Maybe we'll give it a little boop and see if anybody's home.
So there's a question about where we're exploring. Do you want to give an explanation of where you are and where we're going with this expedition?
Sure. Well, where we are right now, we're in the central part of the Great Barrier Reef, on the very outside edge, on the outer edge of the Great Barrier Reef, so on the seaward side. It doesn't look like anybody's home in that shell. Oh, there's someone at home in behind there. What's that orange thing? He's not coming out. He's not. Sorry, we're about 65 nautical miles just northeast of Townsville at the moment in the Central Great Barrier Reef, and we're making our way northward. I think it's a sponge. That orange thing's a sponge. So the title for this expedition is The Northern Depths of the Great Barrier Reef. So we are going to be making our way northward on the outside edge of Australia's northeastern continental shelf, and we're planning to make our way over the next couple of weeks, just about as far north as north goes, as far as we can get with the time available to us anyway. And we're exploring these very remote outer edges of the Great Barrier Reef. Really, this is a this will be the first time that a science ROV has been able to explore these regions, and we're interested in these deeper habitats. Well, today's dive is relatively shallow, but we're going to be exploring the submarine canyons and mesophotic reefs and deep reef platforms. So not the shallow coral reefs that you would typically associate with the Great Barrier Reef that are quite well studied, but we're interested in the deeper regions off the edge of the shelf. This is really, we're thinking of this as sort of the last frontier of the Great Barrier Reef. We'll be heading up off Cape York Peninsula, which is really remote in itself, but then this outside edge of the Great Barrier Reef is even more remote. Very difficult to get to, and it's a real privilege for us to be able to undertake this expedition with Schmidt Ocean Institute and ROV Falkor. You know, we think of the Great Barrier Reef as so iconic, and we've been studying it for decades, and you just about think that we must surely know everything there is to know about the Great Barrier Reef, but no, that's not the case at all. We know a lot about information that we've gathered from a few areas, from the shallow parts of the reef and parts that we've been able to map through remote sensing, things like LiDAR and things like that, but these really deep outer edges are really quite unknown and unexplored. So this expedition is all about exploration and discovery. We've got a whole team of collaborators that we're working with onshore from all over around Australia and also in other parts of the world as well. Each specialists in their own fields, geologists, biologists, taxonomists. We had Dougal Lindsay on the dive the other day where we did one of our midwater plankton surveys. He's our jelly man. So again, we sort of got a few Falkor firsts the other day with Dougal as well. It was the first time that one of these midwater column plankton surveys has been done south of what we call the Capricorn Channel, which is a location in the southern Great Barrier Reef. So that's what this expedition is all about. We're looking at the geology and geomorphology and how these deep features create habitat for all kinds of different communities that you wouldn't see up in the shallow coral reefs. So those are our aims and objectives over the next few weeks. 3 to 4 weeks, and then we're going to come back and make our way back down southward again, and we'll be heading down to Brisbane. We're out here until mid-November, so it's going to be a long trip, and we're going to see lots of new things and make lots of new discoveries, fingers crossed.
Oh, well, certainly new discoveries.
I think, Jeremy, on the last voyage that you and I were on together, I think there was a new something on every single dive that we did, wasn't there?
Yeah, absolutely. Every dive, a surprise. Yeah, really amazing.
So the dive site that we're on today is it's part of what was a massive undersea landslide. We don't really know exactly when the landslide occurred, but what's happened is part of the front of the continental shelf, continental slope in front of the reef front has collapsed at some point, and all of those reef blocks and all this debris has come down from higher up above, and there's quite a large debris field out in front of the face of the reef that we've gone over. We've mapped that previously, and what we're interested in diving on today is we're going to be coming up to the steep face of that scarp of where that that slope failure occurred, and it should be quite steep, hopefully near vertical when we get to the base of it. And we're not quite sure what we'll see there. If we're lucky, we might get to see I guess the rock face of the, if I could say the inside of the reef, so that a fresh face where we'll get to see a bit of the fossil reef underneath. And of course it'll be colonised now by you know, a new community of corals and sponges and invertebrates that will have colonised that now and be using that for habitat. So that's what we're aiming to do today. We'll be passing, well, we're in the mesophotic zone now. We're hoping to be able to get up to around 70 metres, maybe 60 if we're lucky. And our collaborator, Dr. Tom Bridge, who's the coral curator at the Museum of Tropical Queensland, and is one of Jeremy's PhD supervisors. Hopefully he'll be joining us a bit later on in the afternoon for the end part of the dive as well. So we'll have some more coral expertise joining us for that last part of the dive. We've got some specific coral targets that Tom's interested in. This location is quite well known to Tom and his colleagues, but they've never been able to sample these particular coral targets that they're looking for today. So fingers crossed, we'll do our best and we'll have a good look around and see what we can find. It might be another day of some Falkor firsts, maybe some new collections today. Hopefully, fingers crossed.
Yeah, party. I'll cut out and have lunch. But I'll keep watching, and then when we get to the depths where the aquapods of interest are, I'll try to help find some with you.
Terrific. Okay. Thanks, Jeremy. Enjoy your lunch. I hope it was as good as ours.
Oh,
Thanks for joining us.
All right. Talk to you guys soon.
All right. Chat to you later. Little butterfly cod. Hello. It's a cute little one. Do you want to join me in chat? Come see. Oh. What? One fish, two fish, red fish, red fish, red fish, red fish.
Sorry everyone, we were just talking amongst ourselves for a minute there. Sorting out a bit of technology. Another lionfish. Oh, we are rolling around quite a bit here. It's been quite a challenging day for the ROV pilots today. We've got a fair bit of current running. We're bobbing around, Falkor's bobbing around. So it's testing everyone, but we're doing a great job. There he is. Oh, that's beautiful. I've got one of our other scientists at the chair with me here at the moment, Vicky Lowe from the University of Queensland is our third scientist that's on board at the moment, and Vicky's going to be on board for the entire expedition, 47 days, as am I. So we'll see how we're holding together at the end of at the end of 47 days. Yeah, Vicky, would you like to tell us a bit about what your work is and what you work on, and what you do?
Sure. This is very, very far away from what I normally work on. I'm looking at the Southern Ocean and reconstructing past ocean conditions using microscopic organisms called radiolaria, little glass organisms that live right down to well below 3,000 meters. And looking at how the ocean responded over the last glacial cycle, so when it was extremely cold through to when it's quite warm. So it's quite different to what we're seeing here.
And are you sampling by cores? Are you taking cores? Is that where you get your sample material from?
Yeah, so we go down into the Southern Ocean and drill for cores, usually quite deep. Some of them a couple of 100 meters deep. Then what I'm using at the moment is what we call the core tops, and the core tops represent the last about 100 years, and that gives us an idea of what the modern ocean is like, so that when we look down through the core we can compare it and see how past oceans compare to what's currently being deposited on the floor in the last 100 years.
Thanks. We'll keep traveling with you, so that way you don't stop. Yeah, sounds good.
And are you looking at the species distribution of your radiolaria and the community assemblage, or are you doing chemistry on the tests, or both?
We'll be doing a bit of everything actually. So originally my PhD was meant to be reconstructing sea surface temperatures, but to do that I had to go to New Zealand to train, and of course COVID has prevented that training, so I've taken a slightly different approach, and yeah, I'm looking at the ecology at the moment, and community distribution over the entire Southern Ocean and up into the tropics. So actually there are a few core tops around this area that I'm using as well. And how the community changes depending on where in the Southern Ocean it is. So once we've done that, then I will do some down core studies, so looking at past radiolarian communities, and hopefully if I can travel again, I might be able to look at some isotopic work, which is different representations of certain molecules in the tests, and that gives us an idea of what the conditions in the past water was like that those radiolarians were living in at the time. It gives us an idea of how much sea ice there is, what kind of how much water is tied up in the glaciers at the poles. There's quite a few different things that it can tell us.
No, it's okay. No. It's challenging enough.
Oh, thanks, Kerry. Oh, and I've also, I have to say hello to Glenn as well, if he's listening at the moment.
So have you been able to sail down in the Southern Ocean on a research voyage to collect samples as yet, or are you working on material that's already been collected?
Yeah, at the moment I'm working on material that's already been collected. One of my supervisors, he's been working on this data set for the majority of his career, and so I've been able to take that on and have a look at what he's got. A lot of it's already published work, but used for a very different purpose. So, but eventually I'll get down there and collect my own samples. That would be very nice.
It's always been a dream of mine, is to be able to get down, maybe not bouncing around in the Southern Ocean, but at least as close to Antarctica as I can get.
Yeah, yeah. Yeah, it's pretty rough down there.
Yeah. So here we are up in the tropics, just about as far from the Southern Ocean as you can get.
My supervisor is Helen Bostock and Giuseppe Cortesi over in New Zealand.
So we got the opportunity to put this voyage together at very short notice. Everybody, ourselves, the science team, the Schmidt Ocean Institute team onshore and on ROV Falkor have all pulled out all the stops and scrambled really hard in the last few weeks to put this expedition together. So we could really only put together a team of scientists that were already based, certainly within Queensland because of travel restrictions. ROV Falkor has been in Brisbane for the last month or so. So it was fortunate for us that Vicky was available to come along and help us out and get some experience on a research voyage and an extra pair of hands to help us out with all that sample processing that we'll be doing in the lab for Jeremy. So we're glad that you could come along.
Oh, it's been fun so far. I'm sure it's only going to get better the further north we go.
Yeah. We'll see what state we fall off the ship in 47 days when we get back to Brisbane.
Hey, Jared, right off, just gonna be paying in.
Hi Rob, we're seeing your comments coming through on the YouTube. We're looking forward to catching up with you on the 7th when we come into Cairns as well. So we're going to just call into Cairns for a quick stop. We're picking up an extra crew member and picking up 3 more scientists, including the expedition chief scientist, Dr. Robin Beeman from James Cook University in Cairns. And then we will make our way north to Cape York Peninsula and the remote northern outer edge of the Great Barrier Reef. Just seeing a little bit of a subtle change now, I think, in the substrate. Lots of the red algae, rubble.
I just noticed my supervisor Helen is online watching. Hey Helen.
Oh, there. Hi Helen. I was busy looking at one chat feed and ignoring the other one. Hi Helen, thanks for letting us have Vicky for a few weeks. And of course, as much work as we've put into pulling all of this together, it's also thanks to our supervisors and colleagues at work that allow us the time to come away and do this kind of thing. There's certainly a lot of paperwork involved in putting together something like this.
Yes, people stayed up, had very late nights in the week prior to this just to get us on board.
Just saw some halimeda. So the reason I mention that is because halimeda is the organism that I'm working on for my PhD project. So halimeda is a green algae. It's not this fleshy green algae that we've been seeing for ages now. It's a calcareous green algae that grows in little segments, and each segment has got a calcium carbonate or limestone little skeleton on the inside of it, and it sheds those segments, and that then becomes sediment on the seafloor. So this is interesting. What are we now? We're at about 100 meters depth now, and it's getting less sandy and much more rubbly, and everything is coated in coralline red algae, which is the pink, bright pink colour that we can see. Looks like there's lots of sponges in amongst everything there. Now what's that?
Yeah.
Yeah, I don't know. Something interesting there. Zoom in and have a little look. Ah. Is it a anemone or a coral?
Anemone. Yeah.
And an anemone. And an anemone. It's pretty nice. All right. Still seeing lots and lots of this, these cycloclypeus, these flat disc-shaped benthic foraminifera. So, foraminifera are usually quite small, even microscopic at times. So that cycloclypeus is a very large type. It has a photosynthetic symbiont, and that lovely large flat disc shape maximizes its surface area so that its symbiont can get plenty of sunlight for photosynthesis as well. We've got here that an echinoderm with a fish staring at it intently. Don't know what that is. It's a something with tentacles. That is interesting. Any ideas, Luke?
It's got 8 tentacles.
8 tentacles. Octocoral. And what have we here? Is this the pincushion star?
Yeah, it looks like.
Now we're going to get real close and get a look at it.
Oh yeah. Is that what it's called, a pincushion?
Yeah.
Yeah.
Can I touch it?
Yeah. Give it a nudge.
Really good question. What is it? Yep.
You're going to get nothing but encouragement from me.
Someone saying it's the blob star.
Blob star. I think it's a pincushion star. Boop it. You people are incorrigible. Oh no, a pumpkin star. Pumpkin pincushion. Pincushion star. Glenn says pincushion star. Either way, it's very nice. Oh yeah. Oh, okay. That's enough.
Got a little aggressive on that last one.
That was not such a caress and more of a poke. All right.
I've got my whole family watching. Hi Dad, hi Glenn, hi Gary.
Hi everyone in Vicky's family.
Hi.
I hope that my family are out weeding the garden and watering the plants.
Yes, Glenn, have you watered my avocado trees today?
Oh, look at that. Very rubbly. Some little tufts of halimeda in amongst there and sponges and
Another nice star there.
yeah.
Oh yeah, you were.
So we're not really seeing corals here because it's not a hard rocky substrate. But I'm sure that we will get to that pretty soon, and then we'll start seeing more corals. So colourful, it's lovely. Beautiful. Some Leptocerus corals. Wait a second, what's that?
Yeah.
Look at it.
Yeah, let's take a look. Hope you're enjoying your lunch, Jeremy. I don't know if you're still tuned in and watching. We'll do our best to count tentacles and figure out what this is. Looks like it's got a hitchhiker anyway, whatever it is.
Marty, can you just explain the hitchhiker to me?
I was just talking about, I'm not sure if it's a feather star or a brutal star, an extra little critter that is hanging on to that whip coral there. So I just randomly called it a hitchhiker.
And the corals before that Jeremy was saying there was something growing on top of it, and an extension. What's that?
He may have been talking about the zoanthids that colonise a pre-existing substrate, so the stem of something that was there before that's maybe died, and then the zoanthid colony colonises over the top of it.
Right.
He may have been talking about that. Oh, there we go, that's nice.
Yeah.
Are we counting 1, 2, 3, 4, 5, 6, 7, 8? 8 tentacles, so that makes it an octocoral, soft coral.
Yeah, I gotta get up.
Okay.
No, just run. He said he's just gonna run. They just stopped looking for.
Yep.
We're moving on.
Yep. And the other things that Jeremy was also interested in are the associates that live on the corals. So that's sort of a relationship to where certain species of coral have got their associates that tend to live with them as well, and other methods of sampling, whether you take a dredge sample or something, you kind of miss that information. You don't, again, they get tossed around and mixed up, and you don't know which associates came with which coral.
And are those associates symbiotic, or is it more of a one-way?
I'm not sure. I don't know too much about it
Yeah.
at all, really. My guess would be that it's probably a mutualistic relationship, but I'm only guessing.
Yeah.
Are they getting better? Yeah, it's a little bit better, yeah.
Getting a few little platey corals coming in. I think they're Leptocerus. As we come shallower, it becomes a little bit more challenging managing the ROV and the ROV's tether that has floats on it and the ship, and so when we've got a bit of current and moving around a bit like we are today, it's quite a lot to manage, and so we'll see how we go on this dive. We'll keep going for as long as we can. So the question on the YouTube chat about the cameras that we have on SuBastian, is there a forward upward facing camera in addition to the downward facing ones? Yes. There are how many cameras?
We have a total of
10. 10, 11? There are 11, 10. 10 or 11. We have 10 or 11 cameras on SuBastian. Of course, we have the 4K science camera that we are enjoying this vision from. And then there are also a whole bunch of other cameras so that the ROV pilots can monitor what's going on above us and behind us, downwards. Yeah. So whilst we are mesmerized and fixated on one camera, these guys are keeping their eye on multiple, not only multiple cameras, but multiple screens, all at the same time as well.
Jeremy says to look out for the sound camera. They're much better now, Chris. Cool, thank you. I'm looking more stuff now, so I'm done running.
What? Didn't see one.
Starting to approach the depth range now where, if we're lucky, we might find one of these particular acropora coral species that we're looking for. It's a branching acropora, not a platy one. It's got very thin branches, and it will have polyps on the tips as well as all down the branch. Here's a nice fly through of our bathymetry map, the seafloor topography in 3 dimensions that we've dived on today. You can see the little yellow box at the top there is ROV SuBastian, and that's our dive track that we've undertaken today. Wow. This is pretty. No idea what that is.
Oh, yeah.
Might just zoom in a little bit here and take a bit of a closer look. I have no idea what that is. A blue corn dog. It certainly is vivid. It's on a stalk.
It's got no tentacles.
That is interesting. Keeping our eye on the chat there to see if any suggestions come up. One suggestion is an Ascidian. Siphonophore. Tunicate, a stalked tunicate. That sounds plausible. Very nice. All right. So we're really just exploring here. Just have a good look around. We are looking for a particular type of acropora branching coral. And I know that there's a couple of people on shore will be keeping their eyes peeled for that, as will we.
It's another corn
There's
dog.
another corn dog, another blue corn dog.
So the branching coral that we're looking for, how big do they get?
I really don't know, but I would imagine something like 15, 20 centimeters, something like that.
Alright, so just keep on it. Fast. Spin around. Spin. Do report. Yeah. Face forward for sure. Forward. Yeah, we gotta go.
Our ROV pilots are just adjusting our position here a little bit, keeping everything safe. It's been a challenging day. We've got the weather is okay. The weather is not too bad. We've got a little bit of swell on the surface, but we've got currents down here that are making things a little bit challenging.
Still not a good spot.
This is very shallow water for Sebastian to be in, and we're quite close to a coral reef, of course, so that makes it also challenging for the captain and the officers on the bridge of Falkor, keeping us safe.
70 meters below the line. Floats.
We're starting to roll quite a bit up here.
So as much as we have our objectives for each dive, we can only work within the limits of what is safe and what's within the capacity of the vehicle. And when there's current and we're in shallow water, that makes it very challenging. He's beeping.
Starting to go hard show.
And the blue corn dog.
Yeah, those floats are hitting some current right now. Trying to push them away. Roger that. Worker, he's working his way down.
Sorry about that, folks. We are all still here. Just chatting through some logistics. As we were saying, it's quite challenging conditions here at the moment for ROV SuBastian and pilots and the ship. We're getting pulled around quite a bit by this current. We've got floats on the surface.
Cody, I don't know if you're watching the 360 camera, but you can't really pan too much more. Yeah, copy that. There it floats right there.
Push the bus forward. Yeah. Deal with it. Heading off the boats. I'm gonna put 3 or 4 meters until you get close. And I'm getting the extra crazy as it is.
Question on the chat about is the current running parallel to the edge or coming down the slide, which is an excellent question. Seems to be a bit all over the place at the moment, but the dominant current is running more or less parallel to the edge. It's running from the north to the south, towards the south, or southeast. So along this shelf edge here, we are battling with the EAC, or the East Australian Current, which is the reason why we weren't able to dive at one of our dive sites the other day. It was just running too strong, too fast. And it was always going to be a challenge here.
Are you all stopped there, or are you gonna have some more?
I'm gonna hold there for like a few seconds and probably move down another 5 grand or so.
Just see roger that. I got the pinch hole on, water on, so just let me know when you wanna get going.
Yeah. Stretch out there.
Yep, we are rolling around.
Yes, we are. Dazzling
Little
stars.
sea stars down there.
Alright, J-Rob. Hold that right there.
Roger that. Pinch hole coming on.
A lot of this white sediment that we're looking at, it's quite coarse, sort of gravel-sized flakes of sediment. A lot of that is coming from the Halimeda algae that I was talking about earlier. The green algae that's got a limestone skeleton on the inside that looks like cornflakes, and it sheds those. It's got quite a high metabolism, and so it's constantly growing and shedding new segments all the time, and so it ends up being a really prolific sediment producer in tropical reef environments, and that's the white stuff that we're seeing on the seafloor now. Is the skeletons of Halimeda algae. I'm working on, for my PhD project, I'm working on Halimeda bioherms in the Northern Great Barrier Reef. My sites are a bit further north from where we are now, and up on the shallow part of the Great Barrier Reef in the Interreef Lagoon, there's build-ups of Halimeda sediment from all of these limestone skeletons, and they've been piling up there for the last 10,000 years and have created 20 to 25 metre thick build-ups of all of this limestone cornflake deposits that we call bioherms. So that's what I've been working on for the past 3 years or so. Really large features on the interreef seafloor. They create, oh, there's a nice school of fish there. Hey. Are they trevally? The ghosts of Halimeda Pass, that's exactly what they are, yep. Yeah, there's definitely seasonal upwelling along the shelf edge, and that's related to the reason why these Halimeda bioherms are where they are, up on the shallow part of the shelf. They sit leeward or landward of the outer barrier reefs, and it's that seasonal upwelling that delivers cool, nutrient-rich water that intrudes as a sort of a bottom trap layer in onto the lagoon of the Great Barrier Reef, and that delivers nutrients to the Halimeda meadows that then deposit their limestone cornflakes and build up these bioherms over thousands of years. And of course there's Halimeda here as well, where we are right now. This is where we are right now is on the other side of the outer barrier reefs, on the seaward side.
What's
Can you point?
can't see it.
Right. Are we near Viper Reef, Viper Slide? We're just to the north of Viper, I believe.
Passed over that last night.
Yeah. Oh, hi Daniela. I see Daniela's online as well. He's also one of our onshore collaborators. Oh, there we go. We've just thrown Viper up on the inset there. Pilot Chris is having way too much fun right now.
Hey, Jared, I'm gonna pay out a little bit. I'm getting drugged. We're getting sloshing around like a washing machine.
That's an understatement.
Just there.
Oh,
Yeah, there.
there?
Yeah.
Any chance?
What's that?
This?
Oh,
Yeah.
Yeah, right in front of us.
Yeah. There. The white branch thing. I'm pointing at it now on my screen. I can't see it on my screen.
Roger that, you're jumping out.
And board.
Right there.
Alright, my temperature is 55 degrees.
Ooh.
Can't see for very long.
Okay.
Yeah.
Alright, just do what you need to do. Oh. No, I don't think so.
I'm gonna try to block that.
No,
No.
no, it's okay. Thanks.
Alright.
Yep. Do what you need to do.
Yeah, we gotta.
Yep, we gotta go. We're getting hot.
That's a blue thing.
Blue corn dog?
No.
No. What even is a corn dog?
Hot dog wrapped in corn bread. Essentially. Like cornmeal batter.
The best thing on a man.
I had one, I didn't like it. I had one as well.
Sounds
You don't like
terrible.
corn
I only had one, and it wasn't good.
You had it.
At a ballpark during a beer festival. So you think it'd be amazing.
No, you gotta go to Houston Outdoor Rodeo. Big corn dog.
So is it like a Dagwood dog?
Yeah.
Like a Dagwood dog, kind of. Which in New Zealand is a hot dog.
Yeah.
Okay, so then I'll just calm down.
Oh, it's okay. There's a dispute in Australia about Dagwood and hot dogs. So.
Dagwood dog or Pluto pup.
Yes. Yeah.
Yeah, and deep fried on a stick. Dagwood dog, yeah.
Down on my side. Cody.
I think Queenslanders know it as a dagwood dog. Is that fair enough? Oh, Pluto pup.
Pluto pup.
Do you want to point? That?
Or is it the same stuff?
I'm not sure.
No.
It's quite
Yeah, this is a better spot.
What do you want to look at?
We're looking at these quite fine branching things. We're looking for this elusive acropora coral that Tom Bridge has been looking for decades. Yeah. Doesn't look very acropora-ish, does it?
I don't think it does anymore, because it doesn't have the lobate or the plocoid
Okay.
radial correlates.
We've seen some similar things just right in the vicinity of where we are. We maybe just pan around and like kind of stay steady but pan around a little bit. Yeah. Yeah.
Yeah, it does. That's not it. It's
Okay.
should have more branches to it.
Yeah. Any idea what that is?
No. Prozoan.
Bryozoan, sponge, tunicate.
It's prozoan.
Short answer.
I'll try. Okay, we'll look anyway.
Jeremy, that question that you just asked me, we kind of need to move anyway, so we can try. We're just going to go see whether or not we can head down a little deeper.
Yeah.
Head back down to 80. Want to take that question? About acropora.
So Rayrod, acropora or acropora, depending on how you want to say it, it's quite a diverse family or genus of corals. So in the shallows, you get big branching types with like centimeter thick branches that grow meters high, and you also get big plates of acropora, so that form big tables in on the slopes of the reefs. This particular one is one that only grows in the depths where most other zooxanthellate or scleractinian corals don't grow. So in this case, they're quite small, fine, delicate branched acroporas. So with all this fleshy algae around, it's a little bit like a needle in a haystack at the moment. Made more topography.
Yeah. My bad. I thought it was still on. Thought somebody was tapping me in the shoulder. Sorry. There we go. Yeah, I looked up and I saw that. I was like, oh, this was not on. You're good. It's on right now. Let me know when you want to go. Got it.
Fish just dived down a hole.
Updated waypoint bridge.
Are they some kind of wrasse? What do our fish people say? The colour of the acropora that we're looking for, well, the image that we have of it is that it's very light coloured, light grey.
Boat's hard starboard, but there's hard.
He's turning.
Yeah, I don't think it's gonna be quality for only us to do it.
I mean, if he keeps turning his heading like that, I'm gonna have to run the other way.
You're fine right now.
If he keeps turning his heading that way, I won't be.
Yeah, you definitely. We definitely. Yeah, I forgot what string child.
So, yeah. Sort of close out there. It's from the port side.
All these flakes that we can see fluttering around in the water here are those halimeda cornflakes, the skeletons of the halimeda.
We can't get to 80 right now. Mark?
Sorry?
We can't get to 80.
That's alright. Stay where we are. This is the thing we're looking for.
Okay.
Bring it up there first for now. Let's take it. Go down for a bit.
So I know that our colleague on shore from the Museum of Tropical Queensland, Dr. Tom Bridge, is very keen to join us for the last part of the dive today. This is the water depth that he is most interested in at the moment. But I know that he had a very busy day today with a few commitments, so he was going to try and join us towards the end of the dive, which we are pretty much towards the end of the dive. So we'll try and spend a little bit of time looking around for this particular coral that we're looking for. Just so long as the conditions here make it safe for us to do so.
Still need to control him? No, he's got a nice belly right now. We're good. I'm gonna shut down a bit.
Marty, what temperature range does Halimeda like to live in?
Well, we do see, well, obviously in the tropics, and we do see Halimeda down in Moreton Bay, which is off of Brisbane in the subtropics. So I'm not exactly sure what the average water temperature in Moreton Bay would be, but maybe, I don't know, 23, 24, something like that, degrees C. But it's less calcified down there. So, yeah, whether that's a temperature thing in metabolism, I guess.
How susceptible is it to ocean acidification? Is it more or less susceptible?
Well, Halimeda, I guess, as a genus, so Halimeda is a genus, and there are lots of different species. Here in the Great Barrier Reef, there's 14 different species. And so, being a calcium carbonate producer or precipitating a calcium carbonate skeleton, then that does make it susceptible to ocean acidification. There's some papers out where there's been laboratory experiments where they've manipulated the pH and then looked at the skeletal microstructure under the microscope under SEM, and have seen changes in the microstructure at that scale on the skeleton. So it will be impacted by ocean acidification by being able to precipitate less calcium carbonate. So as a result of that, it may become less calcified and more fleshy. But then on the other hand, there's also been experiments where they've manipulated the temperature and CO2, and the Halimeda seems to have done quite well and thrived under elevated temperature and CO2. So I guess there'll be a threshold at some point where it might be conducive up to a certain point, and then it becomes, too hot or too acidic or what have you. So yeah, perhaps perhaps a little less sensitive than coral, but yeah, I don't really know. I mean, there's been times in the geological past where calcareous algae have been quite dominant and more broad distribution than what they have now. So I guess that could be part of these phase shifts that people talk about. We've been in a nice long stable period that's been suitable for coral reef growth for 9,000 years in the Great Barrier Reef now, but it hasn't always been that way. Cute little striped brass flitting around there. We seem to be a bit more stable. How are you doing there? Yeah. Oh, no, we're still rolling around. Yes. Oh, we have two.
There's a question just come through on my phone. How did they get a license for SuBastian?
In the bottom of a cornflake box?
Where did they get a license for SuBastian?
A license to fly?
Yes.
License to hill.
Many, many years of training and experience, I would think.
Actually, you do get certified. You have to have so many piloting hours. Yep, go ahead.
Hey, do you need a break or?
No, we're good, bro.
Sure.
Unless, do you want to come in here?
No, I'm fine with being out here. I just, before you have to fly it up, I don't know if you want to grab a break or anything.
No, I'm good, bro.
Gotcha.
We'll wait until we get on deck.
So what were you saying, Cody? You have to do like
So
like a
many-
logbook of certain so many hours of training, and then get signed off and certified.
Exactly. Get a full flying piloting logbook. At this point, me and Chris don't have logbooks anymore.
Yeah.
Yeah. Because we have so many hours built up into it.
Yeah.
I think my dad would like to get the license. So.
That sea star up there. So we've come down a little bit deeper than where we were 10 minutes ago. 20 metres deeper. We're now sitting at 82 metres. Very rubbly down here.
Yeah.
There he is. That is a cuddly looking sea star. Yeah. Is that a barrel sponge? I have never seen that sea star before.
No.
Yeah. Oh boy. That's just mean. You okay down there? Not so squishy. Squishy? Bouncy. Bouncy, squishy. That is a nice sea star.
It's beautiful.
Lots of different corals though.
That's a sponge. Yeah. It's a vase sponge.
Nice sponge.
Nice little community of fish here right at the moment.
It's a con.
Can't really turn too much to the ridiculous frank.
Oh, that is lovely. It's busy there.
This is the kind of stuff I used to go diving to see, and you never had this long down there, that's for sure.
You're a scuba diver?
Yes.
Me too. Not that I have the time or the money these days.
Not finishing up a PhD, that's for sure.
No. So this is a very nice substitute.
Yes, it is.
In fact, I wouldn't call this a substitute. Shout out if you see something. Grab the winner or something you want to look
Looks
at.
different.
What's that?
Or that one there. That one there.
Oh, yeah.
That one.
The one with the fish,
Yeah.
yeah. Having a fight.
This is a very picturesque little seascape that we're looking at right now. It's beautiful.
Yeah, it's a fungia.
Is that fish guarding it?
Yeah, it looks like it.
What exactly was it that you were looking for?
We're looking at that flat solitary coral.
Okay,
So,
that's
yeah, that one
Was
there.
it the purple mushroom?
Yeah,
Yep.
mushroom coral.
I was in the top.
Oh! The red algae. It's good.
Yeah. Oh, they're so territorial, aren't they?
Very.
Fish on fish crime.
These guys attack you when you're scuba diving. They come and bump into your face
There's a-
mask.
A branching thing up here too.
Do you want to go check it out?
You wanna go? Above us?
Yeah. Just caught my eye.
Good.
There.
Oh.
What is that?
Hold on.
Does it have polyps on the tips?
I think so.
You don't think so?
You want it?
No, I don't think that's the one.
Okay.
Oh, hang on, Tom Bridge is typing.
Good.
Hi Tom, I see Tom Bridge has joined us online. Tom, you can go ahead and come into that Zoom meeting if you feel so inclined.
Just remind him as soon as he turns his mic on, he will be live.
So Tom, if you're joining us in the Zoom meeting, just a reminder that as soon as your mic is on, you will be live on air. So Dr. Tom Bridge is the head coral curator at the Museum of Tropical Queensland. And this is a site that he's been wanting to come and explore for a very long time. So we're here now. And hopefully he can come online and join us for a bit of a chat. Talk about what we're looking at here and help us keep our eye out for this particular species of coral that he's looking for.
Already, as soon as he joins.
Yeah. Okay, Tom, whatever you need to do. Yes, you should see the link. Sorry, everyone out there, I'm having a chat with Tom here. Tom, you should see the link to the Zoom meeting just a little further above us in the chat that we're on right now. John's posted it there. There you go. Playing cat and mouse, trying to find each other in Zoom meetings.
It was 30 seconds too late. So being ended.
Sorry, Tom. It just ended. We're starting another one. Yep. Give us 2 seconds. We're restarting a new meeting. Well, that is nice. Okay, Tom, you should be able to go ahead and join that Zoom meeting when you're ready. And you should be able to see the 4K science camera. It won't be exactly the same as what's going out on the live feed. Sorry folks, we're just sorting out our technical hitches with trying to get Tom to join us via our satellite internet connection. Bear with us for a moment. Okay. That looks like a soft coral.
Okay, there we go.
Hi, Tom.
Hey, how are we doing? You can hear me?
Yeah, we can hear you loud and
I'll
clear.
turn my computer sound off. Probably got an echo. There we go. That should be better. Fantastic. All right. So I'm on my iPhone on Zoom. I'm trying to get onto the. Sorry, I just can't see on Zoom. I can't see the the video feed, so I can get that on.
Okay.
Hopefully I can. Hopefully I can get it on YouTube. There we go. Looks fantastic. Wow, it's a really interesting site. Sorry, go ahead, Marty.
Tom, if you're watching through the YouTube feed, there will be a significant delay between what we're seeing in real time before you see it on YouTube. If you can watch it on the Zoom, that will be closer to real time.
Yeah, I just can't see anything on Zoom at the moment. I've just got your M for Marty, I'm gathering. See anything else on there? I'll try and get in this way. Hang on.
Do you have it now? Are you seeing that now, Tom?
No. Oh, yes, there we go. Fantastic. Yeah.
Great. Okay,
cool.
terrific. That's a good start.
That's looking better. Cool. Okay.
Thank you for joining us.
No, thank you very much. Look, it's super exciting to be on to be looking at this site. This is a site we've been trying to get to for a long time. So it's been a challenge today, but look, this is exactly the sort of habitat we're interested in. So yeah, really exciting to finally see this. So as you're probably aware, we've seen a number of, we're after particularly today some deep water corals that live in this sort of habitat, particularly staghorn corals, which we typically think of as being really dominant in shallow water. So a lot of the tables and the other branching species that you see on most shallow reefs. And they are really dominant in shallow water, but there's a handful of species that live in this sort of habitat here. They're quite hard to see, but there's only a few species, and most of them have only ever been collected once or twice. So they're really, really rare in terms of very difficult to see them, only in dredge collections and stuff like that. But some of the previous work we've done out here has suggested that they're actually quite common in certain habitats. And one of them was originally collected right here, seaward of Bowl Reef by Charlie Varon in the early 1980s, late 1970s even. So yeah, hopefully that's what we're looking for. And this is exactly the right type of habitat that we're in. From what we understand, what we've seen from the AUV from previous trips recently. So yeah, hopefully we'll be able to find it.
So we're in 74 metres water depth at the moment and rolling around. It's 25 degrees C down here.
Geez, yeah, it's pretty warm for this time of year. It's probably pretty similar to up on the surface, but I guess it's, in this sort of habitat, this time of year particularly, these sorts of habitats, there's not much thermocline, so often down to 100 metres or more, unless some sites like Hydrographer's Passage, there's a lot of upwelling. But here we're really in sort of a habitat where I guess, you're often quite similar to surface waters. Once you get sort of later in, early in the year, late summer, you often start seeing a little bit more thermoclines and things like that. So I'm just trying to get that. I'm having some issues with the YouTube feed at the moment. It just goes green. I'm trying to see on my small iPhone and trying to update my. Yeah, it's beautiful. So, is anyone else having issues with the YouTube? Having issues with just about every